So, hello students, welcome to Vedantu NEET English. This is Basuraj sir, your biology master teacher. Students, welcome to the amazing your own channel, right? Now, students, again, good morning to each and every one of you. First of all, good morning, right? Now, students, I believe all of you are set for your NEET journey, right? NEET 2025. Now, in this journey, one thing which is very important and very vital is your mock paper. So, all of you might be aware, right? All of you might be aware, right? Especially every single student from last year, the main drawback of every single student is lack of practice, lack of question practice. Yes, you completely revise your NCRT, you completely drank your NCRT, okay, but as a matter of fact, if you don't do question practice, if you do not do mock test, somewhere down the line you're missing something. So to fulfill that very same reason, we are doing for the free of cost a mock test on your YouTube, students. Now students, I want everyone to quickly tell me in the chat right now if I'm audible and visible to all of you students. Quickly tell me in the chat right now if I'm audible and visible to each and every one of you students. If I'm audible and visible, a quick fire emoji in the chat would be, sufficient for me to know. Okay, students. Now, the rules are very simple. The rules are very simple. Take this entire session, long session, very serious, because this will show your current preparation level. If you are able to solve these questions, you are set, right? If you're not able to solve this question, it means that you need that fine tuning somewhere down the line, students, somewhere down the line. So students, quickly tell me in the chat right now, all of you ready? Yes. Now students, if you're new to the channel, quickly subscribe also. And if you're new to this video, quickly smash the like button by marking your attendance, for marking your attendance, students. Shall we start, students? Shall we start? Yes. Shall we, students? Okay. The first question on your board. Here we go, students. The first question on your board. The first question on the board, and here we go, students. Read the following statements about a cell division and select the correct answer as it goes. Okay, select the correct answer, students. Okay, the M phase represents the phase where the actual cell division occurs, and I is the interface. Yes, I phase represents the phase between the two M phase. So we have one M phase, then I phase, and then M phase. Obviously, right? And this, all of you might be aware that this I phase divided into G1 S G2. True? Okay. Now, statement number 1 seems correct here. In a 24 hours average duration of a cell cycle of human cell, the cell division properly lasts for about only 1 hour. That is, the M phase is said to be only for 1 hour, while your interface is said to be 23 hours is the interface. Okay, makes sense. Okay. Now, the M phase constitutes the more than 95% students? Absolutely not. The M phase is just 5%, while 23 hours, that is 95% of time, is the I phase. So the last statement here is, let us suss here. So last statement is going to be wrong here. So the answer is 1 and 2, and here we go. Option number 1. Done, students? Done, all of you? Done? Students, I want everyone to participate in the class. Students, I want everyone to tell the answer in the chat, because when you answer something, yes, you are attempting the answer. Might as well, students, if you do not know the answer, what is the harm, right? It is not your actual exam, right? So when you try to answer, if you make a mistake, you will learn from that mistake. So make all the mistakes right now in my class, so that you don't have to do the same mistake in your examination. Students, let's go to the next question. Here we go. Second question on the board. Let's go, students. Now, dye injected into a plant cell might be able to enter a adjacent cell through. For example, students, here I have one cell right here. Here I have one cell, plant cell. I have one more plant cell here. So let's say I have two plant cells which are adjacent to each other, which are next to each other here, right? Which are next to each other right here, students. Now what happens is that, right in the middle, all of you know there is something called as middle lamellae. Middle lamellae is present here, made up of calcium pectate and magnesium pectate. Okay. Now, students, what exactly is happening here is the cytoplasm of this cell and the cytoplasm of this cell is connected to each other. Cytoplasm of this cell and cytoplasm of this cell is connected to each other. That is called as cytoplasmic connection. Yes. The other name of cytoplasmic connection is nothing but plasmodesmatal connection. And with the help of plasmodesmatal connection, the contents of this cell can be transferred to this cell. So option number 3 is right here, students. Option number 3 is right. Plasmodesmata. Now, next question on the board, students. Which of the following statements about denaturation given below is correct here? Correct statements. Okay. Denaturation of protein causes loss of secondary and tertiary structures of protein. Okay. Denaturation leads to the conversion of double stranded to single stranded DNA. Okay. Denaturation affects, okay, primary structure which gets disoriented. Start quickly, start students, start. Can we start? I'll upload it, students. I'll upload the notes today, don't worry. Okay. Students, see, this is correct here. This is correct here because in tertiary and secondary and tertiary structure of proteins, whenever the denaturation happens, all the hydrogen bond is broken there. So this is correct here. This also is correct, but students, some of you can anyone tell me why this statement is wrong here? Because primary structure of protein does not have hydrogen bonding. A primary structure of protein is linear in nature. When it is linear in nature, the only bond present there is peptide bond. Only peptide bond is present there, right? So this obviously this becomes a wrong statement. This becomes wrong statement. Okay, this becomes wrong statement. So option number 3 is right. 1 and 2 are correct set of statements here. Next question on the board. Here we go, students. Which of the following areas in India is a hot spot of biodiversity? See, all of you know there is Sri Lanka, Indo-Burma, Himalaya, right? Apart from Sri Lanka province, and then Indo-Burma as well as Himalayan range, there is one more that is present in your South India, students. Almost near the South India. Yes, it is South India, students. All of your entire Karnataka is almost there. That is going to be Western Ghats, students. Western Ghats. Absolutely correct, students. Absolutely correct. Next question on the board, students. Again, the reason I am doing biology questions faster, all of you know the reason. If you're able to solve biology questions faster, it means that you will have more time in chemistry as well as physics. Makes sense? Makes sense. So always try to keep a good pace in biology so you can spend a quality time in your chemistry and physics later on. Next question, here we go on the board, students. Next question on the board. Human placenta is formed by chorionic villi. Yes, makes sense. Umbilical cord, no, not at all. Uterine tissue, yes, makes sense. So answer is going to be both of them. Answer is going to be what 1 and 3, both are going to be the answer here. So students, apart from this, apart from uterine tissue and chorionic villi, there is one more, maternal blood. That also plays a major role in the formation of placenta. Okay, remember that much. Students, can anyone tell me the chart right now? This chorionic villi right here, right? This chorionic villi, on which structure are they growing? Is this chorionic villi growing on inner cell mass? Is the chorionic villi growing on trophoblast cells? In the chat, students, in the chat, answer. Next question on the board, here we go. Okay, good question, students, again. A diploid sporophyte is represented by a dominant phase. So we know a plant where sporophyte is going to be dominant. So algae is removed, bryophyte is removed. Because in algae and bryophyte, gametophyte is the dominant phase, gone. Okay, independent, photosynthetic in nature, vascular plant body. Okay, it alternates with the multicellular, saprophytic or autotrophic, independent but short-lived haploid gametophyte. Okay, now students, all of you know that, see, bryophyte is out of the picture here because not independent sporophyte. Yes, right. Then algae also is removed because here we do not have, sporophyte is not the main plant body. Okay, now the answer is supposed to be between pteridophytes and gymnosperms, right? Now why I will say gymnosperm is a wrong answer, because students, in the case of gymnosperms, the gametophyte is not independent. All of you know, students, here you can see here, right? It says even the gametophyte is independent here, but gametophyte is independent only in the case of your pteridophytes. Only in the case of pteridophytes, students. That is homospory, especially in the case of homospory. Okay, homospory. Next question on the board, students. Next question on the board. Who can get the first answer? Students, I want to see the answers in the chat. I want to see who is the first person to answer, students. Hello, Angela. Hello, Rajkumar. Hello, Shubha. Hello, Kanyaka. Hello, Rajkumar. Yes. Students, is there a lag? I don't think there is a lag. Students, next question on the board. Okay. Next question on the board, students. Which of the following options given gives the correct categorization of 6 organisms according to the type of consumer? Okay. See, all of you know lion is supposed to be in tertiary, so this is wrong statement. A lion is not a primary consumer, no. Grasshopper, cow, primary consumer, makes sense, eating the grass. Fish and wolf, man and lion in the end, yes, makes sense. Grasshopper, phytoplankton, yes, makes sense. Fish and lion, no. Lion cannot be in a secondary consumer. Lion has to be in a tertiary consumer, and the cow has to be in a secondary or primary consumer. The primary consumer mainly, not even secondary. Cow has to be in your primary consumer. So option number D is also a wrong statement here. So option number 3 is right answer, students. Option number 3 is right answer. C for Capto, all of you. C for Capto. Yes. Done. Next question on the board. Here we go, students. In the mitochondrial electron transport system, oxidation of one molecule of NADH gives dash ATP, and oxidation of one molecule of FADH2 forms dash number of ATPs. Very easy question, students. Fairly easy question. Fairly easy question, students. Again, the level of this mock right now, starting mock is around easy to moderate. Easy to moderate is a difficulty level, students. Easy to moderate difficult level. So if all of you enjoy the mock test, if all of you are like, sir, I want to solve more questions, can I get one more free mock test on YouTube for free of cost? So that time, students, we can increase the difficulty level to hard level also. Now this will only happen if you all students watching the lecture, right? I want every single one of you to quickly smash the like button, and also if you're new to the channel, quickly subscribe to the channel right now, students. Then only the second will happen. Otherwise it won't be happening. Okay. Next, students. All of you know one NADH gives how many ATPs? 3 ATPs. One FADH2 gives 2 ATPs. So 3 and 2 is the answer. 3 and 2 is the answer. Yeah, here. Option number 3 is the right answer, students. Option number 3 is the right answer. Option number 3 is the right answer here. Done, students? Done? Okay, students. Next question on the board. Here we go. Students, did all of you smash the like button? All of you tell me in the chat quickly. I want everyone to share this video to your friends also. I want every one of you to share this video to your friends, telling that yes, there is a channel who is doing mock test or live on YouTube for free of cost. Right, first time ever. Right, first and last probably. First and last probably also, if you do not subscribe. Okay. So students, quickly subscribe to the channel and like the video. And also let us know in the comment section. Let us know in the comment section also. Now, the question is about absolute and relative growth here. Now, let me give you a small demo of what is absolute growth here. First of all, listen to me very carefully. Let's say last year, my weight was 60 kgs. In 2024, my weight was 60 kgs. And right now in 2025, let's say my weight is around 65 kgs. Now, in the case of absolute growth, what do? We do something called as final minus initial. That's all we do. So 65 minus 60 is gonna be 60 kgs. 5 kgs is my growth. 5 kg is my actual growth right now. That's all. Okay, 5 kg is my absolute growth. But in relative growth, we do it in terms of percentage. We do final minus initial by initial, right? So final by final. So absolute growth is going to be what, students? See here, absolute is going to be measure and comparison of total growth per unit time. Here, relative is this, because relative is much more accurate, right? It is expressed in common basis exactly per unit initial parameter. That is relative growth. And comparing is this one. So what is the answer? A is 3, B is 1, C is 2. Option number 1. Yes or no, students? Yes or no? Next question on the board, students. Next question on the board. Next question on the board, students. Here we go. Next question on the board. Here we go, students. Quickly in the chat, tell me the answer. Which monosaccharide forms a polymeric polysaccharide that is found in plant pulp and cotton fiber used for paper making? Students, you are supposed to tell me, students. You are supposed to tell me which is the, first of all, tell me which is the polysaccharide we're working with here. We are working with cellulose here. We are working with cellulose. Done. Cellulose done. Now, students, I want all of you to understand what is the monosaccharide. What is the monosaccharide in the case of cellulose? It is made up of long chain of glucose. Long chain of glucose, students. So, long chain of glucose. Okay. Okay, I'll change the question and let's say I will change the question to inulin. If I change the question here, let's say I will ask you the question, tell me which is the monomeric unit in the case of inulin. Then what is the answer, students? Then what is the answer? In the case of inulin, what is the answer? Quick, quick, quick. In the case of inulin, what is the answer, students? In such a case, it's going to be fructose. It's going to be fructose in the case of inulin. Remember that much. Okay. Now, introduction of alien species into a new area poses a threat to extinction of an indigenous species due to, let's say I get a frog, let's say from Mexico, let's say a frog is there, I am bringing this frog to India. Now this frog population will skyrocket now. The frog population will skyrocket. Why? Why, students? Because absence of their natural predator. Because since this frog here, in the case of Mexico, this frog was eaten by a snake. But this snake is completely absent in the case of India. Since the natural predator is absent, the population of invasive species would increase dramatically on the go. Okay, so option number 3 is the right answer there. Next question on the board. Here we go, students. Next question on the board. In ferns, right, students? In the case of ferns, fertilization does not involve archegonia is there, water is there, flagellated anthrazoids is there, pollen tube is not there, students. Pollen tube is not there. Students, all of you should remember, students, pollen tube will come from angiosperms. Sorry, from gymnosperms. Pollen tube formation start from gymnosperm, angiosperm. Only two. Ferns not present. Ferns it is not present, students. Ferns it is not present. Okay. Done. Pollen tube. Pollen tube is the right answer. Next question on the board. Here we go, students. Polyribosomes are aggregation of. What is polyribosome? Poly means many, ribosomes with ribosomes. So multiple ribosomes on one mRNA. For example, students, here let's say we have one mRNA here. We have one mRNA here. On this mRNA, if we have multiple ribosomes on this mRNA, multiple ribosomes on mRNA, that is also called as polysome. It is also called as what, students? Polysome. Okay. So option number 3 is the right answer here. Students, by till now, how many of you are able to solve every single answer? Raise your hands. Or quickly add some fire emoji in the chat if you're able to solve every single question till now. All of you. If you're able to solve every single question till now, quickly fire emoji in the chat. Yes, sir, my confidence in bio is extreme. My confidence in bio is next level because of the preparation which we are doing. Yes, all of you. How is the bio preparation, students? Because biology, oh, I say all of you are crazy doing now. Nice, nice. This is the power of bio, students. Let me tell you again. If you can score about 340 in bio, right, 340 plus or 330 plus also, 340 plus, it's gonna give a major boost to your rank. Now I'm gonna not lie here. So because bio is a game changer, because what happens is that every single time if students take bio as a granted, because bio is so easy, they don't revise biology, and later on in the examination when the bio does not go well, they student cry. Okay, so learn bio, students, learn the bio, learn biology. It's very, very important. 360 marks is not a joke, right? Right, because many students can't even get 360. Many students will not don't even get 360, but you can get 316 only in bio. That's how important biology is, right? And all of you love bio, sure, right? So next question on the board, students. Next question on the board from your chapter, photosynthesis in higher plants, right? RUBP, what is RUBP, students? Is the first acceptor in a cycle? Yes, in C3 cycle. RUBP plus CO2, right? Okay. Pepcase is the product of respiration? No. It is present in mesophyll cell? Yes, it is present in mesophyll cell. In your C4. Phosphoglycolate, students, this is present mainly in your C4 and C3 both, obviously, because in C4 we have C3 cycle, C4 cycle, right? C2 cycle. Let me answer. Phosphoglycolate is a product of food respiration. Yes, it is. So C is going to be 1. C is going to be 1 means this option is gone, this option is gone. Okay. Then students, A is going to be what? A is going to be 3 here, B is going to be 2. Done, students, done. Students, next question on the board. Students, next question on the board, all of you. Next question on the board, students. Match the following. Okay, natural auxin, right? Auxin, ABA auxin. Natural cytokinins is zeatin is natural cytokinins. Then we have natural gibberellic acid is GA3, students. Again, very easy question. Doesn't require much thinking here. That natural auxin A is going to be 2, B is going to be 1, C is going to be 3, and option number 4 is the right answer. Option number 4 is the right answer, and here we go. Next question on the board, students. Okay, in a diploid cell, at which stage of the cell cycle the amount of DNA content is doubled? That is 2C to 4C. When does it happen? Okay, very easy question. Very easy question. Okay, friends, tell me in the chat right now. If I change the question, to, right, if I change the question to, let's say, duplication of centrioles, will the answer change? Will the answer change if I put duplication of centrioles? Will the answer change to some other option here? Absolutely not, because duplication of centrioles happens in the cytoplasm. The answer will not be changing here. Answer won't be changing. It's going to be again S phase itself. It's going to be S phase itself. Next question on the board, students. The yellow round seeded pea plants is crossed with the green wrinkled seeded pea plants. Okay. 4 phenotypes appeared in the progeny in the ratio of 1:1:1:1. Test cross. Okay. The genotype of the two parents are. So in a test cross, what do you take in a test cross, students? What do you take in test cross? What do you take in test cross? Okay. Students, see, this is your F1. So every single time in a test, see, all of you become very smart in this thing now. After the class 12 class, right? Here we can clearly see this is going to be F1 here, right? F1 into recessive parents. Recessive parents is going to be your test cross, and right here, students. Okay. Next question on the board, students. The following graph shows the levels of ovarian hormones during the menstrual cycle. What do 1 and 2 represent? See, if it's the ovarian hormone, you don't have to take FSH and LH. These two are pituitary hormones, right? So ovarian hormones, we have option number 1, option number 4, right? So what is the answer, students? What is the answer? Christina, when will I take plant physiology, students? The next week. The coming week I'll take plant physiology, don't worry. Yeah, all of you are very smart now. Baki, good morning. Okay, see the students. See, all of you know this is happening on the 14th day somewhere here. Here, what happens? Increase in estrogen also happens here. Increase in estrogen happens here, and all of you should know, students. This is called as your progesterone. Why? Because from corpus luteum, progesterone is produced, and this is the increased level of progesterone, and it is maintaining the endometrium layer here, maintaining the endometrium layer right here, maintaining the endometrium layer right here. So it becomes very easy there. It becomes very easy, students. Okay, okay. Now, students, done, done, students. So option 4 is right answer. Next question on the board, students. Next question on the board. Students, target is 400 likes, not 2 or 300. Target is going to be 400 likes right now. Target is going to be 400 likes. So quickly smash the like button, all of you. Quickly smash the like button right now if you're able to answer. Brown algae are characterized by the sense of, students, very easy. Fuco something. What is phycocyanin? Okay, students, there is fucoxanthin. Students, option by mistake. So fucoxanthin. Okay, option number 3. Go for it. Next question on the board, students. Option number 3 is right answer. There was a mistake there. 3 is right answer. Fucoxanthin only. Next question on the board, students. Match the columns of choose the correct option of combination. Time starts now. Quickly go. Okay, students, if you do not know anything, always go for what you know. Always go for what you know, students. See here. Homozygous tall. Here, homozygous tall. Test cross. What is test cross, students? F1 with recessive parents. F1 with recessive parents. Okay. Alleles. What are alleles here? These two are alleles. Monohybrid cross remaining is going to be here. A2R. One like is equal to 4 marks. Nice, exactly. No, 1 like is equal to 4 marks, students. 1 like is equal to 1. They say no. 1 like is equal to 1 pray. They say right in your. It used to come in Facebook before. 1 like is equal to 1 pray to someone. Okay. Now here 1 like is equal to 4 marks. Go. Students, easy question again. Easy question. Easy question. So all of you become very smart now. All of you are above all this. It's very easy for all of you. These questions. Okay. Next question on the board. Which of the following of these is wrongly matched? Chlorophyll contains chloroplast contains chlorophyll. Ilio stores starch. I can see something wrong there only. Amyloplast stores carbohydrate. Yes, amylopectin, amylose is a carbohydrate itself, right? Ilroplast shows protein. Yes, this is also correct, students. This is absolutely wrong here. Why, students? Oil. O for oil. So Elio stores oil here. Okay. Option number 2 is going to be wrong answer here. Option number 2 is going to be wrong answer, students. Okay. Option number 2 is going to be wrong answer here. Okay. Done, students. Next question on the board. Here we go. Okay, they're asking you what is going to be here, A part here and B part here. B part. This part here, this part. That is the B part. I think the labeling has come a little front. So, see, A is going to be the matrix. A is going to be a matrix where we can find ATP formation. ATP formation happens in the matrix. All of you know that. Okay. What does B then? So, only one option is there. Students, by mistake, I told the answer. By mistake, I told the answer, students. By mistake, I told the answer, students. Students, all of you should remember this in your NCRT. Here we have matrix here, entire thing. This part is inner mitochondrial membrane here. Inner mitochondrial membrane, this one. This membrane is inner mitochondrial membrane where everything which we have. So, students, I didn't even see there's only one option there. They should have make the question more difficult. Next question on the board. Students, next question on the board. This is very easy now, students. Again, go. This is very easy. Match the following: Blackman's law of limiting, half leaf, photosynthesis light is dependent reaction, yes. Cornelius Van Til experiment, photosynthesis light is yes. So A is 3 here. Okay. 1 and 4 are gone. What is B? B is 2. Demonstrated that, see, photosynthesis is a light dependent reaction. Student, the question is a little incomplete here. Question, this here, some statement is missing here. Some statement is missing here. Some statement is missing here, students. Some statement is missing here. Can all of you see here? Because Cornelius Van Til told us that wherever the food stores and everything, and so questions, bonus question. Enjoy, all of you. Bonus question, enjoy. Next question on the board. That was the bonus question, students. Exactly, question is incomplete there. Someone ate the question in the midway. They're like very sleepy to type the question. Midway only question went off. Okay. So next question on the board. Apical dominance is overcome by which of the following hormone is against apical dominance, which can promote lateral growth? Which of the following will promote lateral growth, students? Which of the following hormone will promote lateral growth here? Lateral growth. Quick, students. Yeah, bonus question. All of you, everyone loves bonus question, students. See, IAA will promote apical dominance. It's not one. It is going to be cytokinins. I've told you always, students. Cytokinins will promote lateral growth. It is against auxin. Next question on the board, students. Next question on the board. Which of the following events listed below is not observed in mitosis? Not observed in mitosis, students. Not a part of mitosis. Chromatin condensation, yes, it happens. Movement of centrioles to opposite poles, it happens. Appearance of chromosomes, it happens. But students, do you find crossing over in the case of mitosis? Absolutely not. We do not find crossing over in the case of mitosis. Crossing over is completely absent in the case of mitosis. Crossing over is exclusive to the meiosis. Okay, gone. Next question on the board, friends. Match the following 1 and 2. Okay, mitochondria double membrane, lysosomes single membrane, ribosomes no membrane, nucleus also double membrane. So okay, D can be 3 or A can be 3. Okay. Let's take this option for now. Let's say D is 3. D also can be 3, right? Double membrane, right? But here C says 2. C is saying 3, so this option is gone. C is saying 1 here. D is saying 2. Not the option. A is 3. A is going to be 3 here. B is going to be 1. Lysosomes without membrane? No. Lysosome has membrane, single membrane. A is going to be 3. B is going to be 2. B is going to be 2. No. B is going to be 2, correct? C is going to be 1. C is going to be 1. D is going to be 3 again. Students, got the answer, right? Got the answer? Option number 3. Next question on the board, students. Read the following statements and choose the right answer. Calyx is outermost wall of the flower and members are called as sepals, correct? Sepals are usually brightly colored. No, sepals are not brightly colored. So statement number 2 is wrong. Okay. Yes, I told that. Levi Ackerman, that only I told you. Okay. It is supposed to be petals. So statement number only A is correct here. Only A is correct. Done. The purpose of biological treatment of wastewater is what, students? The whole point of sewage treatment plant, the whole point of STP is to reduce the BOD, right? It is to reduce the BOD, biochemical oxygen demand. So STP is done until the BOD is reduced significantly. Until then STP is done. Okay. So answer is option number 1 is right answer. Reduce the BOD. Next question on the board, students. Next question on the board. Next question on the board, students. Read the following statements regarding brow fights and select the correct answer here. They lack true root, stem and leaf. Yes. Male plant body is haploid, gametophyte. Yes. Sex organs are unicellular. Not no. They're jacketed. They have jacket. Fertilization produce an embryo inside the water. Nah. It's inside the female gametophyte, not inside the water. So 1 and 2 are correct. 1 and 2 are correct. 1 and 2 are correct. There we go. Next question, students. Scutellum observed in a grain of wheat or maize is comparable to which part of the seed in other monocot? So it's very easy. Scutellum is a cotyledon, right? Scutellum is a large shield-shaped cotyledon. Large shield-shaped cotyledon in the case of your monocots. In the case of monocots, students. In the case of monocots. Okay. Next question on the board, students. Match the following again from biomolecules. Pigments is going to be what? Pigments is going to be carotenoids, anthocyanin. Okay, A is going to be 4. So these two options are gone. So A is going to be 4, done. Terpenoids is going to be monoterpenes and diterpenes. B is going to be 2 here. C is going to be 3 here, morphine and codeine. D is going to be 1, concalvelinase, lectins. Yes. Friends, do you see that? Even with knowing 2 options, you can easily solve, right? Next question on the board, students. Identify the enzyme 1 and enzyme 2 in the given reaction and select the correct option here. Okay. Time starts now, students. The 2 enzymes in the fermentation process here. The 2 enzymes. The 2 enzymes in the fermentation process. Who can tell me, students? And all of you students, if you're able to answer this question, quickly smash the like button. If you're able to answer this, quickly smash the like button also. Very easy. Pyruvate, decarboxylase, ethinyl dehydrogenase. Easy, students. Easy. Here, what happens, students? All of you should know here. Decarboxylation is happening here. Yes. So decarboxylase enzyme has to be here. Pyruvate, decarboxylase. Then next step is obviously dehydrogenase. Ethanol formation happens. So ethanol dehydrogenase is the enzyme. Option number 3 is the right answer, students. Okay. Done. Next question on the board, students. Natality and emigration. Natality and emigration contribute to increase in population, students. Natality, yes, but emigration, no. Emigration will decrease the population. Imagine someone is going to USA, right, exiting India. Will the India population increase or decrease? Population will decrease, right? Immigration will decrease. Only natality causes the increase in population? No. Natality, right? Natality and immigration. Natality and immigration causes. Right? Immigration causes the population increase. So both are wrong, right? Both are wrong, students. Both are wrong here. Okay. Only natality is not the only reason. It is not the only reason for increase, right? Both are false, exactly. Students, if you're making a mistake, it means that you're not reading the question properly. Read the question properly and then answer one more time. Okay. Exactly. Both are wrong here. Both are wrong. Next question on the board. Oh, good question. Again, A is adaxial top part. Okay. By mistake, I told the answer also, students. Top part adaxial. B is abaxial. C is going to be. Take your time. Take your time. Take your time, friends. Take your time. Friends, take your time. What is going to be the answer? C, D, C or D? Students, see here now. The top part is called as adaxial. Bottom is called as abaxial. Backside, abaxial. Okay. This yellow, small white spot is called as xylem usually. And what is present below here, right? This part, green part is called as phloem here. Okay. If you go back to the question, I told you top part is going to be your adaxial here. Top part is going to be adaxial. Tell me yes or no. Okay. A is going to be adaxial. B is going to be abaxial. C is going to be what? Xylem. C is going to be xylem. D is going to be phloem. Adaxial, abaxial. What is the answer? What is the answer? Which answer, students? What is the answer now? Obviously, option number 2 is right answer. Very easy now. Very easy, students. Very easy. Okay. Very easy, students. Next question on the board. What is lecithin? Lecithin is what, students? Is a glycolipid or phospholipid? Lecithin is a phospholipid. All of you know, students. Lecithin is a phospholipid. All of you know that. Right? Lecithin is a phospholipid. Next question on the board, students. Next question on the board. Select the incorrect pair out of the following. Monadalphus hibiscus mixed in China rose, Diadalphus cucurbita gone there only, Polydalphus citrus, Epipetalis. Okay, Epipetalis is brinjal. Makes sense. Okay, Epipetalis is brinjal. They're asking you which is the incorrect one. They're asking you which one is the incorrect here. Incorrect. All of you should know, students, Diadalphus is only one. All of you should know. Diadalphus is always going to be what? Pea plant. Diadalphus is going to be pea plant. It cannot be cucurbita. It cannot be cucurbita, students. Cannot be cucurbita. Students, if you're able to answer the question, please smash the like button. No. Next question on the board. Here we go. Take your time, read the question, understand what they're asking you. Here, they're mainly asking you about centriole, not cilium flagella. So read the question properly and then answer. Take your time. Students, take your time, take your time. Okay, let's see it, students. Peripheral microtubules, triplet 9 years. Central, there is no. Central, there is no. Okay. Peripheral microtubules, triplet 9. Central microtubules, singlet, no. Peripheral microtubules 9. Central singlet 2, again no. Peripheral 9. Central 0, obviously. Then there is one hub, 9 radial spokes, 9 interdimensional bridge. So 4 is the answer. 4 is the answer. 4 is the answer. Okay. D for dog, exactly. Students, next question on the board. In C4 plants show saturation about 360 microliter per liter, and C3 plants respond to increased concentration and saturation seen at 450. $450 and beyond. This is proper now. Because at 450, no, 450 and beyond. Now, students, can anyone give me an example of your C3 plants which are growing in greenhouse? Obviously, option number 1. Option number 1, students. Now, gametes are pyriform with 2 lateral attached flagella in case. That is, flagella is present here, 2 lateral flagella. Which is that, student? Which one is that? In the case of, where do we find gametes are pyriform? In the case of your brown algae. So, brown algae. In the case of Dictyota, actually, Dictyota. Yeah, brown algae. Dictyota. Okay, tomato and bell pepper, exactly. dictyota. Next question on the board, students. Which of the following are true for cyanobacteria? Okay. So this question little sus. Freshwater, marine and terrestrial algae. Students, here they are considering cyanobacteria as an algae. Remember that. Cyanobacteria as an algae are considering. Okay, so this is correct. No. Algal bloom will happen. Yes. Yes. Heterocyst. 1, 3, 4. Option number 1. Option number 1, students. They will not be present. They often bloom in unpollinated water. No. They'll bloom mainly in pollinated water. That's called as algal bloom. That's called as algal bloom. Okay. Next question about in an area if there are 200 Parthenium plants but a single huge tree with a large canopy, the population density is measured in terms of number of biomass. There's nothing like number of biomass. Number can be used. Number cannot be used that time because number doesn't justify that time. It has to be biomass only. It has to be biomass because biomass of a large tree is more always. It is going to be more always. Okay. So biomass or percent cover. How much of percent cover is happened? Percent of cover is happened. Okay. Or percentage of cover also can be used. Only not number of biomass. It has to be biomass or percentage coverage. How much of coverage is happening? Because all of you know, Parthenium, even though it's very tiny, it is not covering a lot of area here. Right? So, option 3 is the right answer. Next question on the board, students. Phenotypic and genotypic ratio is similar to the case of, that is, phenotypic ratio is equal to genotypic ratio. 1:2:1 equal to 1:2:1. Where do you see this, students? Where do you see this? Where do you see this? In the case of students, in the case of incomplete dominance, students. In the case of incomplete dominance, students. Now, biogas is a mixture of gases containing predominantly carbon dioxide. Biogas is produced by microbial activity and cannot be used as a fuel. Take your time. It's an AR question. Do not go wrong. Students, I have my glasses here only. I can't see the chat. That's why I'm not able to see the chat very well. I'm not able to see the chat very well, students. So students, this is going to be a wrong statement. Assertion is wrong here because the predominant gas is always methane. Tell me yes or no. Predominant gas is methane. Tell me, students, predominant gas is methane here. It's methane. Okay. So the first statement is going to be wrong here, and it can be used as a fuel. So both are wrong here. A phosphoglyceride is always made up of phosphoglyceride. Okay, it's a glyceride, so it has to be made up of fatty acid and glycerol joining together with a phosphate attached. But the question is, student, the question is only unsaturated fatty acids. See, this is gone. This is gone. Okay, because it is glycerol molecule. We can't have glycerol molecule. Okay, it is esterified with not phosphate. Esterified with glycerol, not phosphate. Phosphate is added separately. Okay. Now the question is, students, it's only saturated or only unsaturated or both can be absorbed. Both can be taken. Exactly. Both can be taken, students. Exactly. Both can be taken. Next question on the board, students. Read the following statements and select the correct option here. Last question from my side. Last question from my side. And this is your 46th question. Extra bonus question. Okay. 46th question this is. Read the following statements and select the correct option. A given species may occupy more than one trophic level in the same ecosystem at the same time. Yes, they can occupy. Okay. Productivity of the ocean is less than that of the land. Productivity of the ocean is less. Yes, productivity of the ocean is lesser because in the land we have more and more actual land plants. So more is there. Producers constitute the first trophic level of detritus food chain. No, they do not. So answer is 1 and 2. Okay. Answer is 1 and 2. Okay, students. So that is the end of today's class, students. That's it of today's class. That's all, students. A species can occupy more than one species level. Yes, a crow can be multiple level. What is that? A pigeon can be at multiple levels. Humans can be at multiple trophic level. If a human is a vegetarian, he'll be at second trophic level. If a human is non-vegetarian, he'll be at third trophic level. Right. So, students. Okay. Friend, that is the end for my side today, right? I think now Gopika ma'am will come. Let me give Gopika ma'am a small call and she'll come. Okay, one small call for Gopika ma'am. 1 second, students. 1 second. I am done here. How was botany, students? How were botany questions? Easy, difficult, moderate? Today I gave easy questions, much more easy questions today.
Hello students, am I audible and visible to all of you? Yes? Please let me know if I'm audible and visible. Hi. Can you all see me, hear me? Everything is perfect. Everything is perfect. Chalo. Hello, good morning all the GMAs. So how are you all doing? Hello. Okay, let's start with zoology question, right? In mock paper, we will not have time to tell hello and bye. So let's start directly, okay? Yes? Okay, audio is visible. Okay, quickly share the link with all your friends and let's do the questions of zoology. Okay, ready, steady, go. Please answer all the questions right. Don't make mistake. Okay, be confident that you know everything. Okay, you definitely know everything. It is just a matter of, if you are concentrated to do it or not. Okay, yes, let's start. First question, will it not move like this? Okay, 1 second. Let's just try. Okay, yeah. Don't tell me I have to do this. Oh, okay. Sorry, students. Yes. Okay, first question. How many layers are present in the diffusion membrane of an alveoli? So, what are they talking about? They're talking about the alveoli. Okay, alveoli and your blood membrane. Okay, how does the diffusion happen? If I want to give oxygen, if I want to take in carbon dioxide, what are the layers that are present? And you need to tell exactly the number of layers. Come on, all of you, quickly in the chat box. Okay, come on, answer. Only answers I should see. No talking to each other, no telling hi, bye. Excellent. The answer is going to be obviously 3 layers are there. Can you tell what are the 3 layers also in the chat box quickly? Because we just don't want the answers. We want the explanation as well. Come on, tell me the layers. So here, what is the first layer that is going to be there? Here it is going to be squamous epithelium of the alveoli, right? Squamous epithelium. I'm so sorry, the font is too small. And here what do we have? We have a non-cellular layer, which is the basement membrane. And here what do we have? We have the endothelium, right? We have the endothelium. So question can come this also. They can give you all the 3 layers and they can ask which is right and which is wrong, right? Yes, am I right? Okay, excellent. Okay, so squamous epithelium, basement membrane and endothelium, correct? Perfect. Just give me a minute, students. Just give me a minute. Okay. Perfect. Yes, good job. Moving on to the next question. I hope it will move like this. Okay, yeah. Okay, next question. Answer is done. Which pair of the following belongs to Basidiomycetes? Okay, here they have given the common names. So Basidiomycetes, they are asking biological classification question. Try and answer. If you're able to answer, puffball and caliceps, alternaria and coleotrichum, albigo and mushroom, bracket fungi and puffball. Come on, all of you answer. So this is Basidiomycetes. That means what? They have the basidium, and on the basidium there will be 4 basidiocarps, right? This is the specialized characteristic of Basidiomycetes, right? This is going to be the basidium. So as you are answering, I'll also tell some specialized characters so we can study plus answer. Okay, basidio spores are going to be there. Basidio spores are going to be there, right? Yes. Excellent, excellent, excellent. The answer is the answer is nothing but bracket fungi and puffball. Excellent, all of you. Great job, good going. Okay, proud of all of you. Okay, let's go. Come on, next question. Next question on the next question, yes. Which of the option correctly represents the function of plasma proteins? I told you, if question come, plasma proteins are going to come, and all of you know, out of the blood, okay, 50% is going to be plasma. Not exactly 50, 55% is going to be plasma, and 45% is going to be blood corpuscle. So 55%, we know around 6 to 8% is proteins, okay? And there are 3 proteins that I've taught you, and that is the exact questions that have come here. Come on. Come on, come on, come on, come on. For you, no live has happened. Just refresh and see. Just refresh, it will be fine. Okay? Don't worry, just refresh. Okay? That is why most of you left, I know. Refresh and see. Excellent. What is the answer? Fibrinogen is going to do clotting, globulin is going to do defense, and albumin is going to do osmotic balance. Tell me, what is the serum? Among this protein, what lacks in a serum? Quickly in the chat box. Among this protein, what lacks in a serum? Okay? Yes, sure. Thank you, Akul. I'll check the message. Come on, tell me quickly in the chat box. What is absent in a serum? Faster, faster. Come on, faster in the chat box, quickly. Tell me. Yes, excellent. Okay, fibrinogen is not there, or in other words, we can say clotting factor is absent. Okay, moving on to the next question. All of you are right. Moving on to the next question. Okay, recognize the figure and find out the correct statement. Okay, you have A, you have B, and you have C. Now, according to the statements given below, you have to identify it. A is the fine branch of renal vein. B carries blood towards the glomerulus. C is the tuft of capillaries formed by A. D is a highly coiled network of renal tubules. Come on, all of you answer. Okay, tell me quickly. Tell me quickly. All of you in the chat box. All of you in the chat box, tell me quickly what you can answer. A is a fine branch of renal vein. B carries blood towards the glomerulus. C is a tuft of capillaries formed by A. D is a highly coiled network of renal tubule. I think all of you can answer, right? What is the answer? C. C, yeah? 3 you're telling. C. Students, C is here. This is C. Okay, if you can't see the labeling, this is B. Okay, this is A. And this is D. Okay, D, C, B, A. So see properly and tell me. See properly, check. No. Check properly. Check properly. D is the answer. No. Yes. Very good, Meghana. Very good. I can't say other people's name. Okay. Aishu, Rajkumar. Yes. D is going to be the answer, students. C is Bowman's capsule. C is Bowman's capsule. How will it be the tuft of capillaries? Okay, and B is going to be efferent arterioles. A is going to be afferent arterioles. Okay, the one that is bringing the blood. D is going to be continuing with PCT. No, so it is a highly coiled network of renal tubules. Clear to all of you? So D is going to be the answer. Clear to all of you? Yes, don't make such careless mistakes. Okay, So here see, proximal convoluted tubule, Bowman's capsule, efferent arterioles. So D is going to be a part of the renal tubule, correct? Highly coiled structure. So yes, moving forward to the next question on the screen now. Correctly match column 1 with column 2. So in column 1 you have tetany, osteoporosis, muscular dystrophy, arthritis and myasthenia gravis. All are the disorders that we saw in locomotion and movement. Okay, here we have autoimmune disorder, progressive degeneration of skeletal muscle, inflammation of joints, rapid spasm in the muscle and bone mass decreased. Come on. Yes, come on, answer. Yes, come on, answer. Quickly smash the like button also. Okay, yes, let's make it 400 likes soon. I've already explained what is the mistake. Okay, so just check the diagram. The mistake is that the mistake is there. Okay, yes. 3 you people are telling. Come on, let's solve it. What is tetany? Rapid spasm in the muscles. Can I solve it with 1? Yeah, I can kind of do 50-50. I can strike out these 2 options, right? Okay, let me try osteoporosis, bone mass decreased, right? So B5, okay? C is going to be my deciding factor. Muscular dystrophy, students, progressive degeneration of the skeletal muscle, C2. Yes, it's going to be option number 3. Excellent. Excellent. Okay? Yes, very good. Perfect. Moving on to the next question on the screen, all of you. Yes, next question. Yes. Once the threshold level of an excitable cell, that is neuron, is reached, the sodium gates open and action potential will be generated. The membrane is depolarized. The refractive period is over. Don't worry, I'll teach you neural control. That is, I'll teach you the exchange once again. Okay? Yes. We can solve it with E. Okay, never mind. I go from front to back. You people go back to front, no? Okay, so what to do? What to do? Once the threshold level of an excitable cell is reached, that means once the cell has reached the excitability, so how can you, like among this, which is the first thing that is going to happen? See, other things also are going to happen because you can see the steps are there, but you just tell what is the first thing that is going to happen. Come on, all of you. B for biology by Gopika, Pakka? I doubt. I want all of you to think once again. Once the threshold level of an excitable cell or neuron is reached, what is going to happen? Students, the excitable threshold is reached. So the membrane will become what? Not even one correct answer. Huh? How come? How come? How come? Okay, 2 more seconds for all of you to think. Faster. I don't want any of you to make mistakes. You're capable of answering. Don't listen to your friends. Your friends also don't know the answer. I've always told you this. Someone will tell something, you'll all jump according to that. No, it's not according to that. Yes. Okay, some answers I can see differently, but I need the correct answer. I need the correct answer. Okay. Now some answers are coming. Yes. Tell me. So students, the answer is going to be option number 3. No, the membrane is going to get depolarized. After that, the sodium. See, I'll tell you even this option is right because the sodium gates are going to open. Okay. But the first thing that has to happen is depolarization should happen. Then the gates will open and then action potential will be generated. So we will prefer option number 3 the most. Okay, option number 3 is what is given according to our key answers. Okay. But even option number 1 is correct. Okay. Even option number 1 is correct. So yes, let's go forward with the next question. Next question on the screen. Okay, so I've given an explanation here. So look here. When the threshold level of an excitable cell is reached, the membrane depolarizes, meaning the membrane potential becomes less negative and approaches the threshold potential. This is the first step in generating an action potential. So the correct order will be the membrane is depolarized. I told you the sodium gates will open. Then the action potential will be generated. So stepwise, they're asking you the first step. So I'm not telling that whatever answers you people told is wrong. But the thing, according to the order, this is what first will happen. The membrane is going to get depolarized. That means we are going to reach the threshold. Is that clear to all of you? Yes, I will do that. Okay, I will do that. Yes, Akul, I will see the paper. I saw your message already. Yes, next question on the screen. Clear to all of you. Thumbs up if all of you have no doubts about it. I know the topic can be confusing. We will definitely do neural control. Whenever I get a time, I will definitely make a video on that. Okay. Now, which one of the following is wrongly matched? So we have progesterone, secretory changes in the endometrium of the uterus, aldosterone, sodium reabsorption and potassium secretion, and we know we are going to use this for RAS mechanism. Okay. Oxytocin, uterine contraction and milk ejection, glucocorticoids, emergency hormones of fight and flight. Come on. Yes. Answer. Yes, I will check. I will check. Quickly. Let's also reach the target. Okay. Don't be confused. Okay. We are solving question practice. Marathons are done. Revisions are done. Now question practice we are doing. Okay. Yes. Okay. 4. Perfect. My God. Glucocorticoids are not going to be emergency hormones. We know the adrenaline, that is going to be the emergency flight and fight hormone. Glucocorticoid is going to maintain the glucose level. They also have help in, they also work with immunity. Right. So answer is going to be what? Excellent. Option number 4 is going to be the answer moving forward. Okay. Next question. Easy question. Very, very easy question. Baby question. Okay, PYQs we have already finished. From 2019 to 2024, PYQs are already done. Before 2019, we will do one more session. Okay, yes. Match column 1 and column 2 and select the correct option from the codes given below. So you have amphibians, mammals, contractors, ostrictors, cyclostomata and aves. Column 2, you have air bladder, cartilaginous notochord, mammary glands, pneumatic bones, dual habitat, sucking and circular mouth without jaws. Easy question. Right, I don't think even some of you will take even 1 minute to solve the question. It will take 20 seconds to solve, 10 seconds also. I'll tell you why. Okay, I'll tell you exactly why also. Yes, oh, who is that? Selvan, wrong. Selvan is wrong. Excellent. The answer is going to be option number 3. How did all of you solve? Ma'am, very easy, ma'am. We just answered that amphibians are going to be dual habitat. Automatically, it's option number. Students, match the following majority of the times. One option, neither from the back or from the front, it is going to be correct, and that one option is enough for you to decide the match the following. Okay. So, amphibians are dual habitat. Done, done, done. Mammals are going to have, students, mammary glands. I'm just solving for students who do not know. Okay. Contractors are going to be what? Where do we have? Air bladder, pneumatic bones is there. Okay, so here we have cartilaginous notochord. Ostrictors are going to be, obviously they have presence of air bladder. Cyclostomes are going to be this. Aves are going to be this. Okay, done, done, done. Yes. Okay, very good. Okay. Yes, we do have our DNA batch for all the students who are preparing for NEET 2026. Link is in the description. Okay. Yes. Okay. Even F you can see, even A you can see. Why will you go to F when A only is enough? See, A5. Where else A5 is there? Huh? Yes. One option is more than enough for you to solve that question. Head of cockroach is formed by the fusion of. From where is this? Cockroach. Okay, cockroach is still a controversy because they haven't still approved that insect is cockroach, but still we are going to study it to be on the safer side, because 2024 a lot of questions came from cockroach. They didn't give any question from dengue, chikungunya, families and all that. Cockroach was 100%. Okay. Yes. Exactly. Yes, Subra. Okay, come on, students. Head of the cockroach is formed by the fusion of. Where is our class leader? Class leader, where is the target? Okay, solve the questions. That is more important. Target we'll put later. Yes, yes, yes. A for Ammu ma'am, pakka. Yes, pakka ma'am, because 6 segments. Okay, students, cockroach has a lot to do with 6 segments, 10 segments. So make sure that when you're learning cockroach, I've told you that you have to remember those numbers, right? Because 10 abdominal segments are there, and when you talk about nervous system also, you have to know that, okay, the division is there. Then when we talk about chambers also, there is division. 10 are on the abdomen, 3 are in the thorax. So there's lot of numbers with cockroach. Okay, so always remember all the numbers. So very good. The answer is obviously option number 1. Next one, medical termination of pregnancy, that is MTP, is considered safe up to how many weeks of pregnancy? Easy peasy lemon squeezy question. Medical termination of pregnancy, that is MTP, is considered safe up to how many weeks of pregnancy? 6 weeks, 8 weeks, 12 weeks, 18 weeks. So here again, they can ask you in weeks or they can ask you with the trimesters. Okay, they can ask you with the trimesters. So please be careful of that. Okay. 3. Excellent. 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 Who is that? Who told 1? I'll hit you now. Okay. So what is the answer? Answer is going to be 3. Okay. Now I'm hitting only for you people. No more, oh, Chujo, it's okay, students. One more attempt you have. You take your time. No. From now on, only scoldings. Okay. We have trained you so much throughout the years. Now on, only scoldings for careless mistakes. Okay. Genuine mistakes, fine. Careless mistakes, no. Very good. The answer is going to be 12 weeks. Okay. That's what I told you. They can give you weeks. If it is trimester, students, what will you answer? If it's trimester, what is the number? Can you tell me quickly in the chat box? One student has already put. Excellent. Excellent. If it is trimester, like let's say the same question has come, but they have asked you in which trimester it is going to be. So what will your answer be? Excellent. It's going to be the first trimester. Okay. First trimester. Got it? Done, done, done. Next question. So very big question. Take your time and read. I'll read first for you. Statement 1. The primary lymphoid organ are bone marrow. Okay. Primary lymphoid organ means the military camp is bone marrow and thymus, where the immature lymphocytes differentiate into antigen sensitive lymphocytes. Got it? In military camp, we are going to differentiate them, mature them, origination, all that is going to happen. Now, second statement. The secondary lymphoid organs provide the site for interaction of the lymphocytes with the antigen, which then proliferates to become effector cell. That means this is actually the battlefield. Remember, I told you this is the battlefield and this is the military camp. Come on, answer all of you. Okay. Come on. Any Tamil students? Yes, me. Dandana done is just a, like, there's no purpose. It's just nice word. Okay. Yes. Very good. Very good. Very good. Very good. Very good. What's your answer? 1. Right. Both the statements are correct. Right, this is not an assertion reason, so this does not have to explain this. Many of you get confused with assertion reason and statement based. Students, statement based are individual people, okay? Assertion reason are like family members. One fellow has told something, next fellow has to explain that only. But statement based, they are 2 different individuals. Clear to all of you? Excellent. Moving forward. Yes, I hope all of you are marking your attendance as well, okay? Because we need to make 500 likes. Come on, you people are capable of that. There is a restriction endonuclease called ECO-R1. We know this. What does CO part in the stand for? That means out of ECO-R1, you know this indicates something, this indicates something, this indicates something, this indicates something. I'm going to ask you questions for all of this now. Okay? Yes. Quickly answer, quickly answer. There is a restriction endonuclease called ECO-R1. What does CO indicate? If you tell coelom, no students, I'm going to come now to your place now. Huh? Yes, very good. What's the answer? Coli. Now, what does E indicates? Or tell me, what do the you don't have to tell me it is Eutheridia coli. Just tell me what is it. Is it genus or species name? Because they can ask you any kind of question. Now, this is one pattern of the question. Now, they can ask you, is it genus? Is it species? So, what will you tell? What is this E going to be? What is CO going to be? What is R going to be? What is 1 going to be? Come on, 4 questions here for all of you. Because it is important that not only you solve the questions on the board, you should also make your own questions. Why? What? Which? Okay, 100 questions can come. Come on, tell me. Yes, okay, excellent, excellent, okay. My chats are slow, students, otherwise quality will reduce. That's why there's a delay in chats, okay. Yes, it is going to be genus, species, strain and the number, the sequence at which we found this, okay. Clear to all of you? So this is easier for all of you. They can ask you for hind 2 as well, okay. Hind 2 is Haemophilus influenzae, okay. Haemophilus influenzae, strain is there and 2 is there. Clear? Yes. Very good, very good, very good. Excellent. Moving on to the next question. All of you already answer. Genus, species, not restriction, strain, strain, R strain, S strain, RY strain. Strains are there, no? Like Griffith's experiment, that is going to be this, R strain. Okay, and then I is, 1 is, sorry, not I. 1 is going to be the number, Roman numbers. Okay, yes, excellent. Moving on to the next question. Yes. Use of bioresources by multinational companies and organizations without authorization from the concerned country and its people is called as. So where does this come? It comes in biotech at the last topic. Okay, you might all skip the topic, but let me tell you, one question can come from here. Biotechnology application, last question. Basmati rice they are talking about. Remember all of you? Yes, ma'am, we already remember. Okay. Yes, Roman number it is. Correct. By mistake, I would have told, I, but it is Roman number. Very good. Biopiracy. Very good. Easy question. Okay, easy question. And the rule was found by Indian patent rule, right? Indian patent amendment also was there, but Indian patent rule also came into existence. Okay. Done. Next question. Easy question. Okay, can you see? Let me highlight it for you. They are asking, this is A, okay? This is A, this is B, this is C, and E, okay? D, okay? Yes. Tell me A, B, C, D, E. F is not there, no? Yeah. So they're asking what is A, okay? What is inside B? They are expecting you to answer. Don't tell me it's a flask, okay? C, they are asking. D they are asking, and E they are asking. Oh wow, very nice question. Come on. Yes, British people stole the Indian basmati rice, okay? They made a dwarf variety of it and they were selling it, which is not okay, right? Yes, this is from Miller and Urey's experiment. Come on, quickly. Mark your attendance as well. I want to see all of your attendance marked, okay? Come on. Perfect. Okay, which one are you telling? 1 is it? Ammu ma'am, pakka? Yes, okay, let's see. A is electrode, perfect. Inside this, what is going to be there? Ammonia, okay? All your chemistry knowledge you have to use here, okay? Methane, correct? H2O and H2, correct? These are the combinations given here, okay? Perfect. This is a condenser, this is a U-trap, that is a vacuum. Yes. Option 1 is the correct answer. Let's see. Yes, option 1 is the correct answer. Not option 2, option 1. Okay, not option 2. Because option 2, it is CH2. Okay, what is that? Methyl. Methyl is CH3, right? So option 1 is the correct answer. And C is not spark discharge. Okay, E and D are ulta pulta. Next question. So I've just put the image for those students who have a confusion. Look here, CH4, NH3, H2 and H2. Let me ask you one more question. Spark discharge temperature, students. It has come in 2019 or 18. One question had come about the spark discharge. They have asked about the temperature. Tell me the temperature. I've taught you all the temperature. Come on, quickly. No problem. Don't be sorry. Just correct your mistakes. Faster. What is the spark discharge temperature? You have to know the temperature. If you don't know the temperature, 0. Yes, excellent. It is going to be 800 degrees Celsius. Okay, my charts are a little slow. It's okay. 800 degrees Celsius. Okay, very good. Next question on the screen. Assertion reason question. In humans, there are 2 lungs which are covered by double layered pleura with pleural fluid in between them. The pleural fluid increases the friction on the lung surface, which helps in respiration process. So assertion reason. Come on, answer. Assertion reason. All of you can answer. I hope the slide is clearly visible to all of you. In humans, there are 2 lungs, okay, which are covered by, 2 lungs are there, which are covered by double layered pleura with pleural fluid. So assertion, statement and reason is continuing. Okay, there's no like big difference, okay, like upma and paneer butter masala difference are not there, it's proper, correct. Now you have to tell me if the reason is correct or if the assertion is matching, whatever. Yes, option number 3, excellent. Yes, it does not increase the friction, it decreases the friction and it acts as a lubrication, right? It's like a lubricator. It is for lubrication, okay? Clear to all of you? So that means here they are talking the pleural fluid increases the friction. No, it decreases the friction so that it also acts as a shock absorber. Correct. So it's going to be C. Yes, very good. Next question, all of you. Match the items given in column 1 with those in column 2 and select the correct option given below. This question has come one time as a PYQ as well. Tricuspid, bicuspid, semilunar valves. between left atrium and left ventricle, between right ventricle and pulmonary artery, between right atrium and right ventricle. Easy question, with one itself you can solve, is what I believe. Yes, you can. Yes, lubrication. Okay, lubrication will be done by them. What's the answer here? One, no, don't make mistake, please. Tricuspid, bicuspid. Please remember. Okay, please remember. Okay, this is your heart, right? Between this, something is going to be there, right? Between this and between this. So between this means what? Right atrium, right ventricle, left atrium, left ventricle. Here who is going to be there? This fellow is going to have 3 cusps. Here who is going to be there? This fellow will have 2 cusps. Come on. Yes, excellent, excellent. Okay, some of you first got it wrong, but now all of you are right. Tricuspid valve is going to be between the right atrium and the right ventricle. Okay, bicuspid is going to be between the left and the right. Semilunar valves are present both on the pulmonary pulmonary artery as well as on your aortic arch. But pulmonary artery is going to be called as pulmonary aortic semilunar valves, and aorta is going to be called as aortic semilunar valves. Okay, those difference you have to know. Okay. Heart is so beautiful, huh? Yes, okay, it's going to be between the right ventricle and the pulmonary artery. So it's going to be option number 3. Excellent, all of you. Moving forward, so I've just put the images as well. All this PDF you will be getting on Vedantu NEET English Telegram, as well as in the description box. Okay, as well as in the description box. And if you want to join my Telegram, this is the name, Gopika Biology Official. You will get all the notes of your 11th and 12th, and even this PDF we will share it with you. Okay. Yes, again. So students, if you look here, disorders are all coming as match the following. So disorders is very important for you to study. I will do a separate video. I think I've already done a separate video for disorders. So check that video out. Okay. Match the abnormal condition given in column A with their explanation given in column B. Okay. So glucosuria, renal calculi, glomerulonephritis and gout. Easy question, baby question. Okay. Yes, you will get the PDF on the Telegram channel. Students, we have made it for you. Obviously, we are going to give it to you, right? If the PDF is too big, what do? We put the link in the description and in the comments, okay? First comment will be the link of the PDF. So that way also you can join. Again, 3, huh? Oh my God, today is the 3 day. Okay, what does that mean? It doesn't have a meaning. Okay, what is the answer? Let me try to solve. Can I solve it with 1? Can I solve it with 1? Yes, I can solve it with A alone, no? Okay, presence of glucose in the urine. A is 1 is enough because A4, sorry. No, I need 1 more. So B I have to do. Renal calculi, mass of crystallines. Okay, B3. Yes, done. Yes, very good, very good. Option number 3. Next question. Don't see. Did you all see? Okay. Okay, select, 3 day. 3 day is nice. Select the false statement. During muscle contraction, I band gets reduced. Okay, I band gets reduced. Okay, during muscle contraction, A band remains constant. They want the false statement. During muscle contraction, sarcomere shortens. During muscle contraction, H zone increasing. Okay, I try to cover it with my little hands, but definitely I know all of you saw it, but it's okay. You people are honest people, so I believe in you. Okay, I believe in you. Don't tell, that is not the answer. Okay, sometimes I can put options wrong and all. Yes, during muscle contraction, students, H zone is Hensen zone. In Hensen zone also actually reduces. Why? Because actin will come more towards inside. No, Hensen zone is the place where myosin is going to be there. What is going to happen? Head zone, more myosin region is going to reduce, more actin is going to come in. So head zone also will reduce. Okay, nothing is going to stay constant. The only thing that will increase, okay, for this you will get 0 marks because you saw the answer. Now tell me the only zone that is going to increase. If you answer this, then we will definitely. Okay, come on. Yes, no bonus mark. Now you answer this question. This is your marks. No bonus for all of you. During muscle contraction, dash zone increases. Let's see who is going to tell. Which person? Who is the brilliant person in the class? Come on, faster. Bonus mark. You people are waiting or what? I look like NTA for you to give you 2 more bonus marks. Huh? Okay, very good. Overlapping zone is going to increase. Other than that, all the zones are going to decrease. So now no grace marks. You have genuinely answered. So you are going to get marks. Okay. O zone is going to increase. Moving on to the next question. This clicker is a problem. Okay. It's not clicking on right time. Which of the following structures or regions are incorrectly paired? Okay. Incorrectly paired with its functions. With its functions. Come on. Find it out. They have given hypothalamus, limbic system, medulla oblongata and brainstem. Okay, you will read, no? I will not read. Okay, you read. Take your time and read. Answer. Okay, hypothalamus production of releasing hormone and regulation of temperature, hunger, thirst. Correct. Limbic system consists of fiber tracts that interconnect different regions of the brain and controls movement. Medulla oblongata controls respiration and cardiovascular reflexes. Correct. Brainstem forms the connection between the brain and the spinal cord. Yes, it is going to be option number 2. Limbic system is connected to what, students? All your emotions that you have when you see your NEET paper. Okay, all those emotions, be it the sexual behaviors, all the feelings, whatever emotions we are feeling, it's because of the limbic system. So answer is going to be option number 2. I think all of you did it right. Yes, excellent. Moving on to the next question. Identify A in the diagram. Wow. Identify the A in the diagram. My students wouldn't have even seen this diagram. Come on, tell me. Hypothalamus, hypothalamic neurons. What is the diagram indicating? This is your hypothalamus. This is your pituitary gland. There is an anterior lobe and a posterior lobe. Posterior lobe is the boss. Posterior lobe is like, I will not do with any systems and all. You directly put the secretions into me. That is why this neurosecretory cells have directly stretched into the posterior. Now they're asking you what? Tell me what is this. It's given exactly in your NCRT. Okay. Emotional damage. Sorry, students. Portal circulation in the anterior pituitary, portal circulation in the posterior pituitary, hypophyseal vein in the posterior pituitary, hypophyseal vein in the anterior pituitary. Don't make mistake. I know some of your temptation will be like, okay, let me go for this. Okay, vein I saw. No, exact line, exact image of NCRT. I need exact answers of NCRT. Yes, excellent. Portal circulation in the anterior lobe, right? Because anterior lobe is like, it's okay. You do one thing, you take the help of some fellow. Infundibulum is there, one stalk is there. You take the help of some portal fellow and give me the secretions. But posterior lobe is not like that. Posterior lobe is like, if you want me to do something, you directly add the secretions into me. That is why the neurosecretory cells are directly adding the secretion. That is the story, right? So yes, no B, it is 1. Okay. Portal circulation in the anterior pituitary. It is given in your NCRT, right? NCRT diagram. Next question on the screen. How many of the characters given below are true for echinoderms? What is the trick to remember echinoderms? Let me see how many of you remember. How many of you have seen my video, tricks video? Tell me the answer. What is the trick to remember echinoderms? If you remember the trick, half of the problem is sorted. Faster, faster, faster. Yes, I am from Tamil Nadu, Andhra Pradesh, Kerala, Karnataka. I am this one. What is that? Pan-India person. I am that Pan-India lorry which goes everywhere. No, like that. Yes, excellent. CEO. Okay, CEO. Why I am calling echinoderms as CEO? Because it has starfish. A person who gets many stars is considered as the star of the company. Means what? CEO is the star of the company, right? So yes, CEO is the answer. Now tell me the answer for this as well. Some of you are telling 3. Some of you are telling 2. Okay, very nice. Okay, mixed answers I'm getting. Okay, let's see. An endoskeleton, that is the endoskeleton. Starfish has what? Calcareous ossicles, I've told you. That's why you're not supposed to go and catch the starfish. Okay, they're sharp. Correct. The adult echinoderms are radially symmetrical, but larvae are bilaterally symmetrical. Excellent, perfect. They are triploblastic and coelomate animals. Correct. Digestive system is complete. Correct. Water vascular system they have, not water canal system. Water canal system is porifere. Sexual reproduction, that is internal fertilization. No. Sexual reproduction is most of the time external. Okay. Exceptions are there, but still. And fertilization is not direct. They do have larval stages. Correct. Am I right or wrong? So 5 of them are right, 1 is wrong. So that means we will go with option number 3. Excellent, excellent. Option number 3 is the answer. Next question on the screen. Again, a beautiful assertion reason question. Any doubts in the previous question? No, right? Yes. Huh? Okay, very good. Excretion in cockroach occurs by Malpighian tubules. Reason: each Malpighian tubule is lined by non-ciliated columnar cells. Non-ciliated columnar cells. So we know that excretion in cockroach is coming from, happening with the help of Malpighian tubules. Each Malpighian tubules is lined by non-ciliated columnar cells. That's the question for all of you. All of you answer. You're all capable of answering. Don't worry. Okay, even if it's wrong, it's okay. We are learning. No, I was joking. Okay, careless mistakes, you'll get hittings. Like this question's okay. Yes, answer. Non-ciliated columnar cells will be there. Remember, it is a tubule. So some movement has to happen. So with that general understanding, you can answer. Yes, excellent. If assertion is true, reason is going to be false, right? Because students, again, tubules. We will need in the tubules to have ciliated. Why? Movement is happening, right? So that is why we will not use the word non-ciliated columnar cells as well. So the answer is going to be if assertion is true, but reason is false. Okay. Next question, a big question from molecular basis of inheritance. Read 1 to 5 and find the correct option. So this is how they are going to eat your time. Okay, in NEET examination, they're going to eat your time by giving such big questions. Okay, nitrogen base is linked to pentose sugar, okay, through N-glycosidic linkage. Phosphate group is linked to the 5-OH of a nucleoside through a phosphodiester linkage. They are telling phosphate group is linked to a 5-OH of a nucleoside. Understood? Two nucleotides are linked together by 3,5N-glycosidic linkage. Negatively charged DNA is wrapped around positively charged histone octamer to form nucleosome. Chromatin that is more densely packed and stained darks is called euchromatin. Okay, very easy question. Very easy question. Let me see your answers. All of you tell me your answers. What are you telling? Today is tree day. Tree day. Okay. 3 and 5. Okay. Phosphate group, first one is correct, right? Now, NB joins with sugar with N-glycosidic linkage. If it is a pyrimidine, 1-9, okay, glycosidic linkage. Phosphate group is linked to the 5-OH of a nucleoside through a phosphodiester linkage, okay. 2 nucleotides are linked through, that is wrong. Okay, yes, tell me. C, exactly 3 and 5 are wrong, okay, 3 and 5 are wrong. Okay. Here students, don't get confused. They are talking about the CH2OH. Okay. That is present in the 5-. It is the carbon itself. Just to make you confused, they have given here OH. So don't get confused. Okay. You will be like, ma'am, 3OH we need not to make the bond. Yes, definitely. But here they are talking about how it is going to connect to a nucleoside. Okay. Nucleoside. Clear to all of you? Perfect. Yes. Okay. Yes. Next one, next, students, this is not running, wait, it's moving, it stopped. Did you people tell no, I don't want the answer? Next question on the screen, which is wrongly matched? ICSI, IUI, GIFT and IVF. ICSI, sperm directly injected into the ovum. IUI, semen collected neither from the husband or the healthy donor is artificially introduced neither into the vagina or into the uterus. Gift, embryo with more than 8 blastomeres. We have done this question. I think most of you can remember. Okay. Yes, it's actually a phosphoester linkage here. That is why I told you here this statement, no, it's not completely right. Why? Because a phosphate group really linked to a nucleoside through a phosphoester linkage. Okay, some typo mistake is there. But anyway, that is still right compared to 3 and 5. Okay, clear to all of you? Don't get confused with such questions. Some typos will be there because this we typed, no? So like that. Yes. Gift embryo with more than 8 blastomere stage is transferred into the fallopian tube. IVF embryo up to 8 blastomere stage more than. Come on, all of you can answer this question easily because you know. Because you know the answers. Yes, excellent. Again a tree day. Why? Because gift is not that, no? Gift is where we are taking the gamete from a donor or from that person and transferring it to the fallopian tube. Here IUT should have come. Okay, intrauterine transfer, that is where you are putting more than 8 blastomere stage. Okay, yes, very good, excellent. Moving on to the next question. Cannabinoids, assertion reason again. Cannabinoids are a group of chemicals which interact with cannabinoid receptors present principally in the brain. Natural cannabinoids are obtained from the inflorescence of the plant Cannabis sativa. Easy question again. Easy question. All of you can answer. Did you all mark your attendance? I'm waiting for all of you to mark your attendance. Okay? Don't be lazy. Mark your attendance as well. Okay. Come on. Let me see your attendance. I'm checking the attendance. I hope all of you marked, okay? Don't disappoint me. Come on. No students, come on, let's make it 500 quickly in the chats. Don't be such conjuice, okay? Yes. How many? Okay. What is the answer? 4, 2. What are you people telling? 4, 2, everything you're telling. 2 is the answer, no? If both assertion and reason are true, but reason is not the correct explanation. Cannabinoids are a group of chemicals which interact with cannabinoid receptors present in the brain. Natural, so here they're talking about the extraction, here they're talking about the influence. So 2 different statements. So what is going to be the answer? 2 is going to be the answer, correct? Yes, excellent. Very good. Moving on to the next question. If a person obtained transferment by inserting, oh, very nice question, okay? If a person obtained transferment means what? The plasmid has entered into the cell, okay? By inserting a recombinant DNA within the coding sequence of an enzyme beta-galactosidase, here they are talking about PUC8. He will separate out the recombinant from a non-recombinant by which of the following observations? Very nice question. Very nice question. What happened? Yes, it is okay, Malika. Okay, I'll give you the dates at the end of the class. Okay, I'll give you the dates. It's not that difficult to remember. Just one day before the month end. Okay, don't tell that I told, please. Non-recombinant colonies do not produce any color, whereas recombinant gives blue color. Recombinant colonies do not produce any color, whereas non-recombinant gives blue color. This is what, students, with the help of chromogenic substrate. Remember, that is why I have taught you in biotechnology PUC8. You have to watch my lecture to understand this topic. Okay, biotechnology principles, I have done one lecture where I have taught you about PUC8, where the lag Z is there, and if we do insertional inactivation, what will happen to beta-galactosidase? Okay, yes, excellent. Answer is going to be option number 2. Recombinant colonies will be colorless, they will be white, but non-recombinant, that means insertional inactivation hasn't happened, the desired gene hasn't entered, that will make blue colonies. Okay, perfect. My students are becoming smart day by day. Okay, yes, I've just given an explanation in case someone did not know, but I think all of you know. Next question. For effective treatment of a disease, early diagnosis and understanding its pathophysiology is very important. Which of the following molecular diagnostic techniques is very useful for early detection? This also all of you know. Very easy question. Yesterday itself we revised. Marathon, I taught you this. Early detection. Early detection I taught you. Come on. Answer. Perfect. We have a 1 day. Okay. 3 days we move to 1. Yes. ELISA technique. What is the full form of ELISA? Enzyme linked immunosorbent assay. What is the principle on basis they're working? Antigen antibody interaction. Okay, so for early detection we are going to use ELISA technique. Next question on the screen, very big. Can you see this question? Okay, it's too small actually. Can you see the question, students? Can you please tell me? Mutation, I'll read it for all of you. Match the evolutional concepts in list 1 with list 2. Select the correct answer using the code. Okay, select the correct answer. Mutation, gene flow, natural selection, genetic drift. Change in population allele frequency. Change in population allele frequency due to chances alone. Difference in survival. Difference in survival and reproduction among variant individuals. Immigration, emigration, change allele frequency. Sources of new alleles. I think you can answer. I think you can answer with one option. Not really. 2 options you can definitely crack the answer. Try. You're already telling the answer, huh? Wow, wow, wow. Are you sure? Huh? Yes, ma'am, we are sure. What is the answer? So with mutation, you can see source of new allele, correct, because a mutation happened, a change will happen, right? Variation will happen. So A4, and then we have gene flow. Gene flow is emigration, change of allele frequency. So this 2 is enough for us to find. That is going to be answer number 4. Okay, mutation, source of new alleles, gene flow, emigration and change of allele frequency, difference in survival, genetic drift, change the population allele in the frequency. Clear to all of you? Moving on to the next question. Which of the statement is or are correct? Which of the statement is or are correct? 95% oxygen, okay, 95% oxygen with hemoglobin to form oxyhemoglobin. 20 to 25% of the oxygen combined with hemoglobin to form carboxyhemoglobin. Okay, underline the words carboxyhemoglobin. Each hemoglobin can carry 4 molecules of oxygen. 100 ml of oxygenated blood can deliver 5 ml of oxygen to the tissues under normal physiological condition. That is 100 ml of blood contains 12 to 16 grams of hemoglobin. How many statements are correct? Okay, yes, how many statements are correct? You people are there now again. Again Capto Day, yeah? Let's see. Today is, I think, Shreyas's birthday from for zoology. How many are correct? 4 are also correct. How come? I find one wrong. I find one wrong. 2. Yes. So I have a problem with some questions. 95% oxygen combines with hemoglobin to form oxyhemoglobin. Correct? Yes. 3% is dissolved. 97% is going to form. Correct. But this I have a problem. Is it carboxyhemoglobin or is it carbaminohemoglobin? Okay. Then each hemoglobin can carry maximum 4 molecules. Correct. 100 ml of oxygenated blood. Yes. Okay. 100 ml of this one contains? No. So what is going to be the answer? Option number 3. That is 3 are correct. Right? 3 are correct. Which of the statements are wrong, students? Among this, which of the statements are wrong? Tell me. Let me see if you people know. Which of the statements are wrong? Carbaminohemoglobin, all of you told good. But I want to know exactly which of the statements are wrong. Is it the hemoglobin statement and the carbaminohemoglobin statement? Okay. Quickly, quickly. You should also know which is wrong. Chumma telling answer because someone is telling or you think that his answer is not enough. Right? We need to know the correct answer. No one is answering. Okay, ma'am, got it. Which is wrong? Yes, very good, very good, very good. Okay, next question on the screen for all of you. So here, students, sorry. 4 are right. No, this is also right. So sorry, 1 DL they have given. No, this is right, this is right, this is right. Only this is wrong. Okay, only this is wrong. I'm so sorry. I did not see the 1 DL 100 ml. Okay, this is the option, students. 5 is correct. 5 is correct. Okay. 100 ml of blood will contain 12 to 16 grams. Correct? No? 100 ml or 1 DL of blood. Okay, so that is correct. Only this one is wrong. Only this one is wrong. Okay, so it is going to be option number 3. I thought all of you told 3. Didn't you all tell 3? Did you tell this 3? I thought all of you told this 3. Okay. Yes, 4 options are wrong. Okay, 4 options are correct. Okay, don't confuse. It's okay. Happens. No problem. Okay. Yes. Yes. I thought 3 you people told. Option was 3. Sorry. Confusions like this. The correct systemic circulation pathway. Correct systemic circulation pathway is right auricle, left ventricle, aorta, tissues, veins. Right ventricle, pulmonary artery, tissue, pulmonary vein, left auricle. Left auricle, left ventricle, aorta, arteries, tissues, veins, right atrium. My God, students, you only read. Okay, yeah, 4 are correct. 4 are correct. 4 are correct. Okay. Yeah. Because for me, options are coming very slow, no, for all, like your answers. So that's why I thought all of you told 3, option number 3. I didn't think that you people told 3. Okay. Option 3 only you people told. No, then it is fine. Then it is fine. No, I saw many of them telling 2 and 5. So I just wanted to correct. Okay, very good. Next question. The correct systemic circulation pathway is right auricle. So students, you have to tell systemic circulation. That means you're not talking about the lungs, you're talking about how it is coming from tissues to back to tissues, or like how the deoxygenated blood is going to be carried. Yes. Oh, yes, yes. Quickly mark the attendance as well. Quickly mark the attendance as well. Faster. We have to do next. We have chemistry and then we have physics. 3 is the answer. Okay, you're telling me the blood is going to go to the left auricle, left ventricle, then aorta will take it up, give it to the arteries, then give it to the tissues. From the tissues, again veins, right atrium. Is that the option? Yes, perfect. Perfect. Good job, all of you. Good job. Okay. Yes, very good. Next option on the screen, next question on the screen. Assertion reason. The filtration of blood through kidney is called ultrafiltration. Where the filtration of blood through kidney is called ultrafiltration. Reason. Blood is filtered very finely through the 3 membranes of the glomerular filtration barriers. Come on. Come on, come on, come on, come on. There is no lag. Just check your internet properly. There is no lag. This is not 3, I hope. This is not 3, no? Tell me this one. The filtration of blood through kidney is called ultrafiltration. Blood is filtered very fine through the 3 membranes of the glomerular filtration barriers. What are the 3 membranes, students, here? 3 membranes are what? We have starting from endothelium. Okay, endothelium we have, then we have epithelium, where we talk about the podocytes, and then we have the basement membrane. Okay, in the epithelium we have to remember podocytes, and they are arranged in an intricate manner with filtration slits in between, right? Filtration slits. I hope all of you remember this. Perfect, perfect. Yes, both assertion and reason are true, and reason is the correct explanation for assertion. Excellent. All of you are able to solve assertion. Students, why is it wrong? Here we are talking about filtration. We are talking about ultrafiltration. Here they are exactly explaining ultrafiltration. So don't make the statements wrong. Okay. So you start with endothelium, basement membrane, then epithelium. That epithelium membrane has podocytes which are arranged in intricate manner. Between there are spaces which are called as filtration slits. Okay. Yes. Next question on the screen. The endocrine hormones such as oxytocin, thyroid stimulating hormone and luteinizing hormone are all peptide hormones, tropic hormones, sex hormones released from the pituitary gland. Come on, tell me. This is connected to your hormone, this one. What is that? I forgot. Receptors. Okay, receptors. Easy question, you will all be able to answer. Yes, very good, Tanuj, very good. The endocrine hormones such as oxytocin, TSH, luteinizing hormones are all released from pituitary. Very good, excellent choice, excellent question answering. Okay. Yes, good job, good job. Ayo. This one's answer issue. Okay, this question. Read the following, read the given statement and select the correct option. Okay. Thyroid hormones control the metabolism of carbohydrates, proteins and fats. Statement 2. Thyroid gland secretes a steroid hormone called thyrocalcitonin, TCT, which regulates the blood thyroxine levels. Okay. I hope this is clear to all of you. Question is clear? Thyroid hormone controls the metabolism of carbohydrates, proteins and fats. That is also with basal metabolic rate. Okay. Yes. My birthday? When da? When was I born without my knowledge? Statement 2. Thyroid gland secrete a steroid hormone called thyrocalcitonin. Easily you can tell the answer. Easily you can tell the answer. Come on. Quickly. 2, 3. Wait, statement 1 is correct, but statement 2 is incorrect. Correct. Both statements. No. Think properly. It's again a 3-day. 3-day. Yes, very good. Statement 1 is correct, but statement 2 is wrong. Okay. Correct? Yes or no? Why is this not moving? Yes, this is the correct answer. Okay. Yes. Next question on the screen. Match the following and select the correct option from the quotes given below. Very easy, students. Physalia, Meandrina, Gorgonia, Adamsia. What is this? This is all coming under what? Poha. Poha means coelenterata. Okay, coelenterata. So we know P for Physalia, A for Adamsia. C anemone, brain coral, C fan, Portuguese man of war. Can you solve it with one? No, you have to solve two. Two you have to solve. Okay? Yes. Quickly in the chats. Again Captos Day, pakka. Okay, let's see. Yes. So Physalia, Portuguese man of war. This to everyone knows. So we have only two options, A and 3. I mean, sorry, 2 and 3. Okay, second one. Meandrina is what, students? Brain coral. Correct? So that is going to be option number 3. Very good. Captos Day. Okay. Next question. This is not moving only. Next question. Wow, nice question. Recognize the parts and find out the correct matching. Okay. A is there. B is there, C is there, D is there. Come on, answer. A, B, C, D. A, B, C, D. A, okay, got it. B, got it. C, got it. Yeah, I got it. Did you all get it? Did you all get it? Come on, tell me the answer for this. Did you all get the options? Let me see. It's easy, very easy. Blindly you can answer. Just give 1 minute. Oh, there's something wrong today, right? It should have been Ammu ma'am's day. Okay, never mind. Yes, very good. I'm happy that all of you are answering. This is mandible, which is going to do the grinding part. Maxilla. Maxilla has palps, tasting or sensitivity. Okay. And then C, you have lower lip. D, you have upper lip. Okay. Hypopharynx is there, which acts as the tongue, but they have not asked that. So it is going to be option number 3. Excellent, students. Moving on to the next question. Again, an assertion reason question. Okay. Let me see. Activation of proto-oncogenes under certain conditions could lead to oncogenic transformation. I taught you in the marathon this. Reason, the chemical carcinogen present in the tobacco smoke have been identified as a major cause of cervical and lung cancer. I think this is an easy question you will be able to answer. Come on. Yes or no? Do they have connection or do they not have connection? Yes. Yes, excellent. See, if assertion is true but reason is false, right? So we know about the proto-oncogenes, but here we know proto-oncogenes can get activated with both physical and chemical. Here they're talking about how tobacco smoke can cause, cancer, okay, lung and cervical cancer. So that's wrong, right? Yes, so here assertion is true but reason is false. Next question on the screen. Innate immunity is a non-specific type of defense acquired by person after birth. Statement 2. Physical barriers and physiological barriers are a part of innate immunity. Easy question. All of you will be able to answer. I got 3 again. Are you people sad at this point? Huh? Come on, come on, come on. Easy question. Okay, yes, faster. I think this is around, we have done around 40 questions already. 42 questions are already done, I believe. Okay, innate immunity is a non-specific type of defense acquired by a person after birth. Yes, very good. Again, it is an option number 3. That is going to be statement 1 is incorrect, but statement 2 is correct. Students, what to do? It's coming in that option. It's not like I'm wantedly doing, na? No. Think properly. Why are you people telling option number 1? Both the statements are correct. Innate immunity is non-specific type of defense. Okay, but acquired is by birth. From when on? Acquired is acquired by a person after. Oh, no. Wait. Innate immunity is a non-specific. Okay, I thought acquired by, not by a person. Okay. Innate immunity is a non-specific type of defense. Acquired by, acquired. No. Students, this is a continuation. Okay, comma is there. No, this is not about acquired immunity. They are saying innate immunity is a non-specific type of defense acquired by a person after death. I mean after birth. English problem this is. Okay. This is a single statement. I thought it is acquired immunity and I thought it is not. Okay. I think most of you would have gone confused with the English here. They should just put this in a small letter. Okay. Acquired by person after birth. So this is going to be the answer. Okay. Statement 1 is incorrect. Statement 2 is correct. When I saw many of you doing 1, that's when I thought there's something wrong with English. Okay, this is a single statement because it's a comma and it's not a full stop. I need to take grammar classes at this point. Okay, yes. So it is going to be option number 3. Done. Innate immunity is acquired by birth, not after birth. No, not after birth. Yeah. Obviously, Supriya. Then why are we doing question practice? Time pass, huh? This question that follows has 2 statements. Reason is supposed to be the explanation for assertion. So here assertion goes like this. A bacterial cell with no restriction enzymes will be easily infected and lysed by a bacteriophage. Reason, restriction enzymes catalyzes synthesis of a protective coat around the bacterial cell that can prevent bacteriophage attack. Okay? Yes. English is killing me, not me killing English. Okay? Yeah. Who is that? Yeah? Sauparnika. I'm not killing English. English is killing me. I thought, okay, they're talking about acquired immunity. I'm a person who thinks very normal, basic. After innate immunity, I thought, okay, acquired immunity. Then I realized there's a comma and that it's a continuation. Okay? What to do, students? Things like this keep happening. Yes, A is true, but R is false is what all of you are telling. A bacterial cell with no restriction enzymes will easily get affected and lysed by, because the restriction enzymes are molecular scissors and those people are capable of doing what? They are going to cut, okay, restriction endonucleases to be specific. They are going to cut the bacteriophage genetics so that bacteriophage will not affect them. But all this is nonsense, right? They are not going to do protein code and all of that. So what's going to be the answer again? Okay, it's going to be option number 3. Okay. Match the following columns and select the correct option. You have BT cotton, ADA, deficiency, RNAi and PCR. I have taught you this very recently. Okay, students, we are almost coming to an end of the question. I'll just inform Diksha ma'am as well. Okay, just give me a minute. I think we are left with like 3-4 questions. Okay. Oh, Sandesh. Yes, very good. I haven't told all the students' name who is telling correct, right? Huh. Okay, let's see. BT cotton, Bacillus thuringiensis, A4. Only one option is there. Still, we will go forward. Adenosine deaminase deficiency, students. Cellular defense in eukaryotes. Okay, in eukaryotes. RNAi is the gene therapy. Okay. No. Sorry. I'm scratching more. Okay. Okay. Once again, BT cotton, Bacillus thuringiensis, correct. And adenosine gene therapy, RNAi, cellular defense and PCR this. Okay, so done. Yes, it is going to be option number 3. Very good. Next question on the screen. Okay, I think these people were lazy when they were typing. They were like, okay, let's just put all the options 3 only. Okay, but don't do that for everything. The figure below shows 3 steps A, B, C of polymerase chain reaction. Select the option giving correct identification together with what it represents. Okay, A, B and C is represented. So you have to tell which among this is correct. Okay. No, not again. Okay. Come on. Tell me the answer for this. Students, one question at least is going to come which does not have, okay, that answer. Come on, come on, come on. Answer quickly. Let me see, some of you are asking some doubts. Okay. That's all. We are almost going to have 46 questions done. Okay. Correct. Okay. Very good. Very good. Yes, Saubarnika, they are peptide hormones, okay? Oxytocin and TSH, they are peptide hormones, okay? Yes, 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 yes. They are peptide hormones, but the question is related to from where they are released. No? Again, students, answer is going to be C, extension in the presence of heat stable DNA polymerase. Here, instead of DNA polymerase, they can give you Taq polymerase as well, okay? Taq polymerase as well. Okay, moving on to the last second question on the screen, okay? Yes, the insecticidal protein in Bacillus thuringiensis does not kill the bacteria itself because, read it, okay? And I hope all of you have marked your attendance as well. Come on. Quickly tell me. Quickly tell me. Call your name. What? I look like one attendance person or what? That I'll call all of your names. Students, come on. We need 20 more likes and we'll reach the target. Okay? LH is not. Sauparnika only asked TSH and oxytocin, no? LH is not. Only TH and oxytocin she has asked, or has she asked LH also? No LH. LH is not. Sorry, I did not see the LH only. Since I can't see the messages so far, okay? Even in the phone, I can't see it that fast. Like the messages are going really fast. Right? Teen ka din. Okay, yes. Teen ka din is going to be... Yes, the toxin is present in the inactive form and gets activated upon entering an insect. Okay, yes, very good. So it will get activated because of what, students? Because of alkaline pH, right? Yes, we are coming to the last question and I hope Diksha ma'am is ready as well. Okay, last question for the day here. In PBr322 plasmid, role of ROP. Role of ROP is codes a restriction site, codes an antibiotic resistant gene, codes for the origin of replication, codes for the protein involved in plasmid replication. We are breaking the teen ka din, okay, with the last question. Come on, all of you, in the chat box. Come on. Last question for today. So every single day from today, you are going to have practice, be it mock papers, be it AIMS level, JIPMA levels, questions, PYQs, like that. Many things are planned for all of you. All you have to do is stay subscribed to the channel and follow every single video where we are going to solve questions. Okay, yes, we have broken, finally 3 days are being broken into 4th day, that is going to be codes for the protein involved in plasmid replication. Okay, yes, give me 1 minute, students. We will just connect with Diksha ma'am. By then, quickly smash the like button and make it 500 likes. One second. Shall I do? Okay, I'll just tell the students. Keep your phone silent. Yeah. Akshay, phone. Yeah. So now Diksha ma'am will be joining for chemistry. After that, you will have physics. Okay. So don't go anywhere. Stay here only. Okay, no problem. Mistakes happen. So definitely let us know in the comments how the session was. And for those students who are looking for offline centers or online batches, all the links are in the description. You can always check it out. Okay. Yes, now Diksha ma'am will come. I am leaving. Bye bye. Take care, all of you. Have a great day.
Hi students, welcome to the class, and now it's turn for chemistry questions. So everybody let me know in the chat box if I am clearly visible and audible to you, so that we can start with very, very interesting questions of physical, organic and inorganic chemistry. We are surely going to fall in love with these questions, and this is a very good start to your practice, okay? Because theory parts we have already covered, right? So everybody, if you're ready for the chemistry questions, then let me know in the chat box and fill this chat box with green hearts, right? With green hearts and fire emojis, fill this chat box, right? So that we can start with the first question on your screen. That is, which of the following statement is incorrect? So first question is regarding the statement here. So one reaction is given to you. If you see this reaction, what is this? Gabriel phthalimide reaction, isn't it? So we know that the product P after this is going to be 1 degree amine, that is C2H5NH2, because this is the halide that you have here, right? Now tell me in the chat box, what is this reaction? What is the name of this reaction? Where you have CHCl3 and NaOH. That reaction is carbylamine reaction. So answer will be C2H5NC, right? Now, if you see product P reacts with grignard reagent to form hydrocarbon, okay, gives reaction with benzene, definitely. What is benzene sulfonyl chloride? Anybody? You have, this is your primary amine, right? This is Hinsberg reagent. So Hinsberg reagent definitely gives right answer with the, it means it gives soluble product with the 1 degree amine. So that means this is the correct answer. Product Q is good smelling. Not at all. Q is isocyanide, that is foul smelling. So incorrect option they were asking. That means C is the incorrect option here. That means C is the correct answer, right? Next is product formed in the above reaction. So this is the reagent that is given to you. This is happening in the presence of NaOH and water at 100 degrees Celsius with several hours. That means the temperature is very high. That means it is going to break those bonds even which are difficult to break. So what will happen here? This bond, firstly, it will break, right? This bond will break, this bond will break. This is Na positive, this is OH negative. So what will happen here? Wait a minute. OH, no, this bond will break here. This bond will break, this bond will break, this bond will break. Here you will have attack of OH negative on this electrolyte. No, wait a minute. This bond will to break afterwards. Firstly, though, this is your nucleophile. This is a base. But because there is no alpha hydrogen, acidic hydrogen, that is why this is going to act as a nucleophile. So OH negative is going to attack here. That is why when it will attack here, this will eliminate the oxygens here. Same thing will occur on this side, right? And it will eliminate the electrons here, right? That means oxygen will be eliminated. Same oxygen will attack here and electrons will be shifted here, right? That means over here you will have 1, 2, 3, O negative, O negative, O negative, right? Okay. Then in the solution you also have H2O. That is why that will be added here. So you will have OH, OH, OH. That means this will be your one of the products. Rest is, this will give rise to R, C double bond O, O, right? And this is H. Now, why they have written RCONA here, ma'am? That means RCONA will not be formed. And should I write B as the correct answer? No. Because in the solution, you have NaOH, right? So that means easily Na positive can replace the the H negative here, H positive here. So it will be RONA. That means this is also the answer. So answer is both 1 and 2 are formed here, right? Both are formed here, right? Next question is the mole fraction of solvent. They are asking the mole fraction of solvent in aqueous solution of solute is this. Mole fraction of a solvent in aqueous solution of a solute is this. Okay. Mole fraction of a solvent is given to be 0.8. Mole fraction of solute will be 0.2. Whenever you have something like this, what you take? Take number of moles as same as mole fraction. Number of moles of solvent is 0.8. Number of moles of solute is 0.2. Now, if you assume this thing. If you assume this, are you getting the same answer of mole fraction of 1 and 2 the same? Yes, because total number of moles will become 1, right? So that means we can assume that. Now, what is the molarity? Molarity is equals to number of moles by weight in grams, then it will be multiplied by 1000 here, right? So here you have number of moles. This is weight of solvent, so this will be N2. If you multiply this by M1 and divide this by M1, then can you say that this is nothing but your mole fraction of 2, right? So here, and in mole fraction, you always put this weight in grams, right? So here, that is why I multiplied it by 1000. So this is, sorry, in number of moles, you always put this in grams, right? So that is why I multiplied it with 1000. This is N2 by N1 multiplied by 1000 divided by M1. Now N2 you have, N1 you have, right? 1000, what is M1? Aqueous solution, so M1 will be 18. So answer is going to be, directly put the values and you will get your answer here, right? Okay. Directly you just need to put your values here and you will get your answer here, right? Next question is, you have a compound where you are adding I2, NaHCO3, major product they are asking you, right? So see, firstly you need to know the function of I2 and NaHCO3. Bicarbonate you have here. So firstly, what will happen? I2 will react on the pi bond, okay? So it will react on the pi bond here in the electron densities and it will form a cyclic intermediate. A cyclic intermediate will be formed here, right? Okay. Then here you will have something like this, okay? This is your ring and here you will have, this is I, this is I, this is the thing that you will have and it will break. This is C double bond O, this is OH, right? Now you have a bicarbonate, right? You have a bicarbonate. Bicarbonate, it is like a base only. It will try to abstract the proton from here, right? So it will abstract the proton from here. And what will exactly happen? This O negative is going to attack here because this is partially, it is very weak. Why it is weak? Because it can easily lose its electron to this iodine, right? So that is why what will happen here, you will have, this is the ring and here you will have I and here you will have 1, 2, this is C double bond O and here you will have the attachment of oxygen, right? This is the kind of answer that you will have here. So I, this is double bond, that is not there. Here you don't have I. You should have I over here, that is only in B. So answer is B is the correct answer. Is that clear, everybody? B is the correct answer, right? Okay. So I have picked up really good questions for you. So don't worry about that, right? Okay. You will have a good practice. Now, this is a very interesting question, okay? Try to see what exactly. So, why this is interesting? Because some of you will answer this in very high confidence that I answered correctly. But I'll give you one hint that there is some rearrangement involved here. There is rearrangement involved here. Okay. Now, let us see what is the rearrangement that you will see here, right? So this is benzene OH triple bond and then double bond OMe. So first thing is you have Lindlar's catalyst that is going to attack on alkyne, right? What will happen? This will break one of the pi bond and will attach hydrogen here, right? Firstly, that thing will happen. After that, you will do hydrolysis. See what exactly is going to happen here. Here you will have two. Here you will have double bond, single bond, OH. This is the thing that you will have as A. After that, when you are doing acidic hydrolysis, now in that mixture you also have acid, right? That is the reason, right? What does acid do? If you have OH, it will try to stabilize the OH. It will form first, it will get converted to carbocation. It will eliminate the OH here, right? So what will happen here? Here you will have this formation that will be there because of this Lindlar's catalyst that is done. Right? Here it will be H2, okay? H2, not H2, right? So here what will happen when you add H positive in the acidic condition, this OH will be eliminated, isn't it? OH will be eliminated, guys, and this is the carbocation that you will have here. Now, very important thing, there will be rearrangement of carbocation. Now you will tell me why. Here you have a benzene ring which is attached to a carbon positive. So there will be resonance. Okay, there will be resonance. If it undergoes rearrangement, right? If it undergoes rearrangement, then this pi bond can shift here, this pi bond can shift here, and hence you will have a positive charge over here. That means you will have a formation of this kind of product, right? Here you have carbon with positive and here you have OMe. Now tell me why we are rearranging. Ma'am, here you had resonance. Ma'am, it was stable only with resonance. Then why you need to rearrange it? What is the reason? The reason is back bonding. The reason is back bonding, right? If you remember one rule that we studied in GOC, that is BARHI, Barhi. That is back bonding has more priority than resonance. This R is the resonance, this is back bonding. So that means back bonding will win, right? So that is why this is a more stable carbocation. Now in that solution, in that mixture, you have water also. Water has H positive and OH negative also. So now what will happen? Because it is acidic hydrolysis, that is why H positive will firstly eliminate the OH negative, convert it into carbocation. Then in that solution, you also have OH negative from water. So that will attack on this carbocation and you will have the formation of this thing, that is CH, OH, and this is OMe will remain as it is, right? Here you will have the addition of OH, right? After this, you are again adding H positive. So now see what will happen. This H positive in the acidic condition, it is going to replace this methyl group with hydrogen. So here you will have one carbon which has two OH. OH negative will be eliminated, H will be eliminated, and this bond will shift here, and hence you will have the formation of C double bond O, and H is already there, so it will be CHO. That is why this will be converted to, as you can see here, this will be converted to CHO, right? So that is going to be your correct answer here. That is C is the correct answer. Is that clear to everybody or not? Is that clear? Okay. Next question is, which of the following species is not paramagnetic? Now anybody, which of the following species is not paramagnetic? Okay. So paramagnetic, guys, very simple. Paramagnetic, not paramagnetic means they are talking about diamagnetic. Okay. Now firstly you have to tell me one thing. Wait a minute. Yes. So now you have to tell me one thing. If you have MOT for less than 14, less than or equal to 14, I'll just say less than or equal to 14, you have 1, 2, 2, 1, right? For more than 14, you have 2, 1, 2, 1, right? Is that right or wrong, everybody? Yes? Okay. Okay, I think I accidentally wrote it opposite. So this will be 2, 1, 2, 1. This is 1, 2, 2, 1. Okay. Now let us solve these questions here. This is having 5 into 2, that is 10 electrons. This is having how many electrons? 7 plus 8, that is 15 electrons. This is having 8 plus 6, that is 14 electrons. This is having O2, that is 16 electrons. Right. Now here what you will see, 10, 15, 14 and 16, right? Now one trick here for you. Chalo, trick I'll tell you later on, right? First thing is this B2, if you talk about this B2, 10 electrons means it is less than 14, so that is going to be 2, 1, 2, 1. That is why when you fill the electron, always fill the electron minus 8. 10 minus 8 because 8 electrons are in 1s and 2s bonding, anti-bonding, right? So that is why this is 2 electron, this is 15 minus 8. How many electrons to fill? 7 electrons to fill. This is 14 minus 8, that is how many electrons? 6 electrons to fill. This is 16 minus 8, that is 8 electrons to fill here, right? Okay. Now if you see, this is 15, so it will be 1, 2, 2, 1. This is 14, so it is 2, 1, 2, 1. Wait a minute, I'll just write it like this. This is 2, 1, 2, 1, right? This last one is 16, so that is going to be more than 14, so it is 1, 2, 2, 1. Okay, so that is how we are going to draw this, right? 1, this is the second one, this is the third one, and this is the fourth one. Now let's fill the electrons here. 2 electrons, 1, 2. It is what? Not paramagnetic. It is paramagnetic, right? Unpaired electron it has, no? It is paramagnetic. It is 7, 1, 2, 3, 4, 5, 6, and 7. Again, it is paramagnetic, right? Now fill this 6 electrons here. 1, 2, 3, 4, 5, 6. It is diamagnetic. Diamagnetic. 16 minus 8, students, that is 8 electrons. Again, fill this 1, 2, 3, 4, 5, 6, 7, 8. Again, it is unpaired electron. That means it is paramagnetic. That means the answer is going to be diamagnetic. They are asking you. CO is the diamagnetic compound here. Is that clear to everybody? CO is the diamagnetic compound here. Right? Okay. Now, let's see the next one here. The next one is, which of the following is incorrect for electron affinity? Electron affinity, see, electron affinity of alkali metal is positive, right? That means affinity means that they want to gain the electron. So metals wants to lose the electron. So their electron affinity is positive. That is correct, right? Electron affinity, they want to gain the electron. They want to, basically, if you see, metals are, alkali metals are like M positive, right? So they are electron deficient. So that means they have the tendency that they want to gain the electron, right? Inert gases, they have fully filled orbital, right? They don't want to gain the electron. So their electron affinity, they don't want to gain, their affinity will be negative, right? If you forcefully give them the electron, their electron gain enthalpy will be positive. I'll just write it here. Electron gain enthalpy is positive. Electron affinity is negative. Why? Always these signs are opposite to each other, right? Your want to eat something, if it is positive, I want to eat something, when after you eat it, you will feel happy, you will release energy, right? Electron affinity is your want, and this is after eating something, you feel happy or sad. So if inert gases have negative, they don't want to gain the electron. If you forcefully give them the electron, they will not release energy, they will absorb energy, right? Okay, so that is, it is negative. Oxygen has lowest electron affinity in its group. That is also true. Can you tell me the reason? Because if you talk about oxygen, it has small size. Because of its small size, and it already has 2 lone pairs here, so it does not want to gain the electron. So it's lowest EA in the group. That means even though it is very electronegative, then also it does not want to gain any electron, right? Electron affinity of polonium is half of Cl. That is also true. So incorrect answer is EA of inert gases is positive. That is incorrect. Next is correct order of hydration enthalpies of alkali metal ions. Alkali metal ions, the correct order of hydration enthalpy. See, hydration enthalpy, if you see now, we know that smaller the ion, if the ion is very small, then more, see, I'll just give you one example. It's like running, okay? So if someone is very slim, right, easily they can, you give them one, like, one mukka, one punch on the face, they'll be like, ooh, right? So they can easily doze off, right? So what will happen here, right? So basically more molecules can attack if it is very small, if someone is very thin. If someone is so big, Everyone will be so scared of him or her. Like, oh my God, so dangerous. I don't want to punch him. So, not other molecules, other water molecules can get stick to that molecule. So, that means if your size is small, if lithium, let's say, has very small size, so more hydration will be there, more hydrated molecules can attack there. That is why it is going to be have maximum hydration enthalpy. So as you go down the group, hydration enthalpy decreases. So answer is going to be, this is the correct answer. Size of ion is inversely proportional to size of hydrated ion, right? And it is, if size of hydrated ion is large, that means hydration enthalpy will be large. Hydration enthalpy. Hydration enthalpy. Right? So A is the correct answer here. Next is correct order of ionization enthalpy of group 14 elements. Group 14 elements, everybody. Group 14 elements. Correct order of ionization enthalpy. Ionization काशी गए संत प्रभु. Ionization enthalpy has opposite trend in here. काशी गए संत प्रभु नहीं. It is काशी गए प्रभु संत. Okay. It should be काशी GESNPB. But tin and lead will interchange each other. So answer is going to be A is the correct answer for ionization enthalpy of group number 14. The most commonly used reducing agent. See, here it is Al3 positive. There is no logic behind that reduction and oxidation. Pb2 positive. So it is SN and then it is Pb. Right? If you talk about the group number 40. We know that lower oxidation state is more stable as you go down the group. That means SN is more stable. Pb is more stable in 2 positive state as compared to SN2 positive. Right? So here if you see Pb is in +2 state, that means it is chilling. It is already in +2 state. Here Sn is in +4 state. Here Sn is in +2 state. Now we know that as we go down the group, lower oxidation state stability increases. That is why what it will try to do, SN4 positive will try to undergo SN2 positive. That means it will try to undergo, no, actually what you will see, SN2 positive will try, because this is more stable, no? So this will try to undergo SN4 positive. That means it, because here higher oxidation as compared to lead, higher oxidation state is more stable. So it will try to undergo SN4 positive. That means it is undergoing oxidation. That means it is if it is undergoing oxidation, then it is very good in reducing something else. It is a very good reducing agent. So answer will be D is the correct answer, which is a commonly used reducing agent, right? Next is SnCl2. If you have seen this in organic chemistry also, we have used that in Stephen's reduction, if you remember, right? Among XeO3, XeO4 and XeF6, the molecules having same number of lone pairs on XeR. So here you should know the diagrams, okay? So if you talk about XeF6, XeO3, let's draw these diagrams, okay, one by one. Same number of lone pairs they're asking you which will have, okay? So if you talk about XeO3, right? Simple thing is xenon is belonging to which group? Xenon belongs to group number 18, 8 electrons. Xenon is bonded to 3 oxygen. That means 6 electrons are already occupied. It has 1 lone pair. 1 lone pair plus 3 bond pair, 3 sigma bonds. When you are calculating hybridization, you have to calculate only sigma bonds. So 1 lone pair plus 3 sigma bonds is going to be 4. That is sp3. Right? So that means xenon is 1 lone pair and these are the, this is the geometry. So you can write it here 1 lone pair. They are asking you same number of lone pairs on xenon. So what is Xe? What about this one? So if you talk about second one, that is XeO4, in this case, what you have is 8 electrons, and you know, it is forming all the 8 electrons here. All the 8 electrons, that means 0 lone pair it will have, right? It is having 0 lone pair, no lone pair, no lone pair. What about XeF6? Xenon F6, we know that it is having 8 outermost electrons, 6 are involved, so it will have 1 lone pair. That means 1 lone pair is there. So XeO3 and XeF6 has 1 lone pair, that is same. Right, so answer will be B is the correct answer here. Wait a minute. XeO3 does not have a lone pair. XeO3 has a lone pair. XeF, XeO3 I have drawn, XeO4 I have drawn, XeF6 I have drawn. XeO3, XeO4, XeO3, XeO4, XeF6. No, they don't have the similar lone pairs. Okay, so answer will be B is the correct answer. Okay, I think there is some typing mistake here, because here they have taken this example as well. Right, here they have taken this example as well. So that example can have 1 lone pair, right? But this has one, this has one. XeF4 does not have any lone pair, right? Okay, ma'am, can you please tell us why XeOF4 has lone pair? Let's do this. Okay, so XeOF4, xenon belongs to group number 8, right? 2 bonds are there for oxygen, 4 bonds are there for fluorine. So it is -6 is equals to 2. That is 1 lone pair. So definitely it is going to have 1 lone pair. And its geometry is 1 lone pair plus 4, 5 bond pairs. So it is going to be 6. That is sp3d2. That is why it is existing as the octahedral geometry, where 1 lone pair you have on the octahedral, one of the octahedral positions. Okay. So here the answer will be, here the answer will not be this. If they write it here, XeOF4, then I can say the answer is D. But here they have written XeO4, that is why I will regard the answer as XeO3 and XeF6. So I'll just delete this option here. Okay, this page here. So answer is going to be B is the correct answer. Okay. Next we have, which of the following orders are correct as per the properties mentioned? Okay, as per the properties mentioned, you have to see which is correct. Let's say acidic strength, right? Acidic strength, as you see, phosphorus and sulfur. Okay, this is phosphorus, this is sulfur, right? Right, and here you have silicon, and here you have arsenic. Right, so if you see here, arsenic and this are in the category of amphoteric. Definitely, as you go to this side, acidic strength is non-metallic character. If you see, na pi usse, yes, okay. SO2, this is P2O3, right? And they are asking you correct. Chalo, I'll just, I don't think so that is the correct one, because over here, acidic strength increases, okay? So, because non-metallic character increases, so SO2 should have more acidic strength as compared to P2O3. Okay, let's see this one. Enthalpy of vaporization, okay? This is nitrogen, phosphorus, arsenic. Down the group, more negative electron gain enthalpy. This is also not correct. Okay, enthalpy of vaporization, it is the opposite order. NH3, vaporization means, right, to get vaporized very easily, to get converted into gases, right? But that is not possible in NH3 because it has hydrogen bonding, right? So it will not have maximum enthalpy of hydrogen vaporization, right? This is also not correct. This is also not correct because SO2 should have more as compared to this. Talking about more negative electron gain enthalpy, more negative. Now, if you see fluorine and chlorine, that is only correct. Chlorine has more as compared to fluorine, because fluorine has very less electron affinity. It does not want to gain the electron, right? So that is why more electron gain enthalpy, the tendency to gain in, the electron affinity will be more for chlorine, and that is why it will have more negative electron gain enthalpy. That is incorrect, right? Here also it is incorrect. Oxygen will have less, sulfur will have more. Again, because oxygen has small size. So this is also incorrect. Last is thermal stability. That is down the group stability will decrease, right? Why we are saying that? Because size of the atom is increasing, so bond length is increasing. It is easy to break that. That is why D is going to be the correct answer here, right? Okay. Yes. Next is the correct order of decreasing second ionization enthalpy of titanium, vanadium, chromium, and manganese. Okay. Correct order of decreasing ionization enthalpy. Second, they are asking you. Yes. So if you see this titanium here, it is vanadium, okay, chromium, then you have manganese. This is Steve, that is 22, 4s2, 3d2. It is Steve, 4s2, 3d3. Steve, Cur, that is 4s1, 3d5. Manganese is 4s2, 3d5. After losing one electron, what it will have? Okay, after losing one electron, it is 4s1, 3d2. It is 4s1, 3d3. It is 4s0, 3d5. Maximum we already know, guys. This is the maximum. Can you tell me the reason? Chromium will have the maximum second ionization enthalpy. Can I see any way which is chromium is maximum? That is over here. Why? Because it is very difficult to take electron from a very stable electronic configuration. So that is why answer is going to be A is the correct answer. Okay. Next is amylopectin is composed of which thing? Amylopectin is composed of which thing? Anybody in the chat box? Amylopectin is composed of two kind of linkages, glycosidic linkages, right? That is C1 and C1C4 and C1C6, right? These two linkages are there, and in this case you have alpha glucose. So answer is going to be C is the correct answer. Okay, understood? Next you have among the following the incorrect statement is. Incorrect statement they are asking you based on the linkages again. Cellulose and amylose have 1,4 glycosidic linkages. That is correct. Okay, amylose has 1,4, amylopectin has 1,4 and 1,6 both. Right. Then lactose contains beta D galactose and beta D glucose. That is also correct. Maltose and lactose has 1,4 glycosidic linkages. That is also correct. Okay, maltose and lactose both will have 1,4 glycosidic linkages. Sucrose and amylose have 1,2 glycosidic linkages. Here see, sucrose will have 1,2, but amylose has 1,4. Okay, so here answer will be D is the incorrect answer. Okay, answer is going to be D is the incorrect answer here. Incorrect statement, that means the correct answer. Okay, glucose and galactose are having identical configuration in all the position except for which position? See, there is only one position, that is which position? Anybody? They are epimers of each other. What is that? C4 position. Over here, if you see, it is opposite. All the other positions in glucose and galactose are same. Okay, so that is at C4 position, they are epimers of each other, or you can say cyclic epimers and anomers of each other. Right. Next is magnetic moment. Let us try to calculate the magnetic moment here. Magnetic moment of 2.84 Bohr magneton is given by which of the following? Anybody in the chat box? Magnetic moment of, we are saying magnetic moment of 2.84 Bohr magneton is given by which of the following? So atomic numbers are given to you. This means you should have number of electrons should be equals to 2. See, if you have 1 point something, number of electrons should be 1. 2 point something, number of electrons should be 2. 3 point something, number of electron 3. 4 point something, number of electron 4. 5 point something, number of electron 5. Okay. So here what we will have, we know the electronic configuration, right? Okay. So for titanium, we know it is 22. So answer is going to be 4s2, 3d2, right? Chromium is what? 4s2, 4s1, 3d5. Cobalt is 27, so it is 4s2, 3d7, right? Nickel is 28, so it is 4s2, 3d8. Now, we will take out how many electrons? 3 electrons from here. It is going to be 3 electrons from titanium, guys. It is going to be 3d1, right? 2 electrons from here. It is going to be 3d4. And 2 electrons from here. It is 3d7. And 2 electrons here. It is 3d8. Now, what we will do, right? So, here what we have is 2 unpaired electron we need to have. As you can clearly see, it has 1 unpaired electron, it has 4. It will have 3. 1, 2, 3, 4, 5, 6, 7. So 3 unpaired electron. It is 1, 2, 3, 4, 5, 6, 7, 8. So that is having 2 unpaired electrons. So answer is very good. Nickel 2 positive is the correct answer. That is D is the correct answer here. Next question is, which one of the following does not correctly represent the correct order of the property indicated against it? Okay, does not correctly represent. Okay, let us see that. Increase number of oxidation states. Okay, this is increasing magnetic moment, increasing melting points, increasing ionization enthalpy. See, very simple. This we have just done that. Out of this, chromium will have maximum, because second ionization enthalpy means you will have to take out electron from 3d5, which is very difficult. So it will have maximum. Over here you will see something weird. Manganese, if you remember in the graph I told you here that manganese and technetium, they both have a solid dip here. They are the minimum, they have a dip, they are abnormally low melting point, right? So, and now what they have written here is highest melting point here. So that is definitely not possible. So answer is going to be, this is the incorrect. Okay, that's what I told you in the 4 hours marathon of class 11 and class 12 chemistry, right, that we have done. Why that was important? Because these some kind of things that I have told you directly from your NCRT that are going to be very important for you, right? Next is, given below are 2 statements. 2 statements are given to you. All the following compound react with para-toluene sulfonyl chloride. That means Hinsberg reagent. So this is your aromatic. This is basically aniline. Okay. This is 2 degree amine. This is 3 degree. Aniline is 1 degree. So aniline will react. 2 degree can react, but it will give insoluble product. Okay, with the alkali it is insoluble. 3 degree will not react. So this statement is false. This statement is false. Okay, this statement is false. Next is, the products in the above reaction are soluble in aqueous NaOH. Is it all the products? No. Only the product of primary alkali, only the product of primary amine are soluble in the NaOH, right? So answer is going to be, this is also false. So answer is, both the statements are false. Very good. That is D is the correct answer. Next we have, okay ma'am, here you have benzene, you have CN, you have COOCH3 here, right? You're reacting this with Grignard's reagent. So what is going to happen, guys? So this is CN, this is C, double bond O, this is CH3. So what you will see here, that here this is positive, this is negative, this negative is going to attack here, and hence these electrons will jump here. So this carbon will, what it will have? This carbon will have O negative. After hydrolysis, this will get converted to OH here. So I'll directly write OH. This is CH3 and here one CH3 will attach. Okay, so 2 CH3, 1 OH will be there. So answer is, wait a minute. Acha, I forgot. Oh, I forgot CN there. I'm so sorry, beta. I forgot CN. CN was also there, na? Okay. So sorry. Huh. So this is C triple bond N. Grignard reagent will also going to break the pi bond over here, right? Okay. So how it is going to react? So this this R negative part is also going to attack here. Okay. So I'll write it there. It is also going to attack on this nucleophilic position. Electrons will jump here, right? Here what you will have, C double bond N. This will be MgX here. Here they have given MgBr, so I'll write MgBr. This is CH3. I'll just write CH3 here. Yes. Now, after hydrolysis, right, just the way this OMgBr was converted to OH, this will be converted to double bond O, and hence you will have COCH3 here. So answer will be D is the correct answer. Okay, D is the correct answer here, right? Next we have, for a dilute solution, or they have given you Raoult's law states the following things. Okay, Raoult's law states what? Now sometimes they give you also, it's a mixture. Mock test means mixture of difficult, easy, all the questions. Okay, so this is one of the questions that all of you can easily answer. Try to answer this. Yes, everybody. For a dilute solution, Raoult's law states that the lowering of vapor pressure is equal to mole fraction. The relative lowering is equal to mole fraction of solute. Relative lowering is equals to mole fraction of solute. That is what relative lowering. Wait a minute, dilute Raoult's law. They're not asking you the, wait a minute. Vapor pressure solution is equals to mole fraction. The relative lowering of the vapor pressure is proportional to, I think, chalo theek hai. The vapor pressure of the solution is equal to mole fraction. Yes, so answer will be this. Relative lowering will be equals to this, right? Okay, that is your relative lowering in the vapor pressure, right? Raoult's law is different. Raoult's law is when vapor pressure is equals to vapor pressure in of that component in the mixture, that is P, is equals to P naught into mole fraction. This is what it states. Okay, let me see if that statement, vapor pressure of the solution is equal to the mole fraction of the solvent, that is also not given here. Chalo, no issues. Out of this, so we can answer that only. And azeotropic solution of two liquid has boiling point lower than either of them when they are saying boiling point is lower. There is decrease in the boiling point. So you see, very simple thing. If you increase the boiling point, there is elevation in the boiling point, that means vapor pressure will decrease. If there is increase in the boiling point, that will occur when you will have positive deviation. See, if you have positive deviation, what will happen? Raoult's law will fail definitely. Your total vapor pressure will be greater than vapor pressure of A plus vapor pressure of B. If your pressure increases, right? If this is the atmospheric pressure, you have to reach atmospheric pressure. Pressure, we are saying earlier it was this. So you need to have this much boiling point. Pressure is increasing, so you need to have less boiling point. That means there is lowering of the boiling point. Okay, so that is why answer will be C is the correct answer, right? Next is you have one numerical based question where you have NO2 required for reaction is produced by decomposition of N2O5 in CCl4 as per the equation. This equation is given to you. Initial concentration of N2O5 is given to you. It is initial concentration of N2O is this, and it is this much after 30. Means initial and final concentration both are given to you. Rate of formation of NO2 they are asking you. It is quite simple. First, you should know the rate of reaction. That when you write it in terms of N2O5, it will be, it is disappearing. Na? So it is minus 1 by 2 d of N2O5 divided by dt, right? In terms of this we can always remember when you're writing rate of reaction, you will divide it with the units. Unit means stoichiometry. You will divide it with the stoichiometry, right? This is the stoichiometry, ma'am. Plus 1 by 4 d of NO2 divided by dt, right? That is the first thing that you should know, right? Second is, ma'am, okay, we know this. Now, after this, what we know, right? They are asking you the formation of NO2. What is the appearance of NO2? In appearance, you don't divide it with the stoichiometry. That means appearance is D of positive of, because there is appearance, so this is equals to this. So if you want to equate this value, that will be equals to minus 4 by 2 d of N2O5 divided by dt. So can I write here minus 2? So that means all you need to calculate is the rate of change of d of N2O5 divided by dt. You multiply it by minus 2, you will get your answer, right? Okay. Just give me a minute. Let me see. Appearance of N2, that should be positive, right? Okay. So now initial concentration of this is given to you. This is also given to you. Time is also given to you. So what you can do, you can calculate the value of dN2O5 divided by dt directly by final minus initial. Final is 2.75 minus of 3. You will say, ma'am, this is negative. See, this has to be negative to make this thing as positive. Just give me a second, right? Divided by 30 minutes. Okay, if the answer is in per minute, you don't need to do any conversion here. Just divide it with 30, right? Now you solve this value. This will come out to be in minus. Put this value here and you will get your answer positive. Okay, understood, students? You just have to solve this value of delta of this. You will solve this value and this is what you will get here. Then you will put this value here, right? So basically this is your, if I put this is 0.25, so I'll write this here only, right? So this will be 0.25 divided by 30. I'm just going to write it because, you know, I know when I write myself, then you feel that, okay, ma'am, the question was easy. Otherwise, you feel that, ma'am, the question was tough. Now, I'll put this value here, right? That is d of NO2 divided by dt is equals to minus of 2. And what we have calculated, d of N2O5 by dt is minus 2.5 by 30. So, this is minus 0.5 by 30. That is nothing but this is 15. So, you have to calculate this value, that is 0.25 divided by 50. Okay. After calculating this, you will have your answer. Okay. After calculating this, you will have your answer. That is going to be, this should be positive only. This will be in positive. So, answer will be plus 1.667 into 10 raised to power minus 2. That is going to be your answer. You can calculate for yourself and see what is the answer that you are getting. Next is the most abundant element by mass in the body of healthy human adult are oxygen, carbon, hydrogen and nitrogen. Okay. The weight of which is 75. Now they are saying that a person who has 75 kilogram weight. What do you think? What is my weight? I am pointing at myself. Do you think I am 75 kilograms? Right? Do you really think so? Okay, let's guess ma'am's weight in the chat box. Guess that. Right? Not at moon. I am talking about on earth. Right? Okay. Not at moon. Right? So here it is 75 kilograms. That is the weight of the person. Most abundant. Now they are saying that 10% of it is hydrogen. That means in a body, in that body, how much hydrogen? 1 will be there. 10% of 75. That is nothing but that is 7.5 kilogram is hydrogen. Why I have calculated only hydrogen? But the question is about hydrogen. The weight of which this person would gain if 1H, that means isotope 1 is replaced by deuterium here. So if you replace it with deuterium, replaced by deuterium, then it will have 7.5 multiplied by 2, because this isotope number 2 has double the mass, so it will be multiplied by 2. That means it will have 15, right? 15 kilogram, right? That means the difference gained by person, the amount that is gained by person is 15 that it has now because of this H2. Earlier it had 7.5 of hydrogen. That means 7.5 gram of kilogram of it has gained. So answer will be C is the correct answer. Is that clear to everybody or not? Is that clear? Okay. Next we have is 1 gram of carbonate on treatment with excess of HCl produces this much mole of CO2. The molar mass of M2CO3 in gram per mole. Let's see that here. So basically you have M2CO3 that produces CO2, right? Okay. If carbonate on treated with excess of HCl, okay, excess of HCl, it produces this much mole. So that means if I see this here, if 1 gram of carbonate on treatment with excess of HCl, this much mole of carbon dioxide molar mass they are asking. Okay. Firstly, you should know the equation here. If you see the equation here, this is M2 and this is 2 of MCl, and you have only 1 mole of CO2. That means 1 mole of M2CO3 is giving you only 1 mole of CO2. Right? From there, how I will solve? This is 1 mole. This is also 1 mole. Right? If I see this here, if 1 mole of M2CO3 is giving you 1 mole of CO2. Okay, firstly I'll calculate the number of moles. That means 1 mole of CO2 will give you 1 mole of M2CO3. If 1 mole of carbon dioxide is produced by 1 mole of M2CO3, then 0.01186 mole of CO2 will be produced by 0.01186 mole of M2CO3. That means here number of moles is equals to mass by molar mass. So molar mass is equals to number of moles into molar mass, wait a minute, into mass. Mass is given to be 1 gram. This number of moles is given to be 0.01186 multiplied by 1. So that is going to be your answer here. And when you solve this, this is in grams, this is in moles, right? And they're asking you in gram per mole only, isn't it? Why the answer? We got the answer wrong. Wait a minute. Given mass by molar mass, isn't it? Oh, ma'am. Ma'am, if we replace it here, molar mass is equals to given mass by number of moles. Oof, ma'am. Oof. What you are doing, ma'am? Huh. I think I'm a little bit tired. So number of, if you see, number of moles is coming out to be 0.01186 and mass is 1. Now you will see your answer. That will be there. Okay? Understood? So answer, what we have calculated is coming out to be B. Okay? So I have just solved unitary method. Totally your choice which method you want to choose. Unitary method is like best for everything, right? So next we have is number of radial nodes of 3s and 2p are respectively. So number of radial nodes is n minus l minus 1. For 3s it is going to be 3. What is s? S, P, D, F. 0, 1, 2, 3. So for s it is l is equals to 0. This is minus 1. It is 2. So if you talk about 2p, it is 2 minus 1 is equals to 0. So answer will be 2, 0. The answer is A is the correct answer here. Okay. Next is the hybridization of atomic orbitals of nitrogen in all of these N2O positive, NO2 positive, NO3 negative, NH4 positive. See, firstly, we all know that NH4 is going to be sp3, right? So you have this is eliminated, this is eliminated. Next is when you talk about NO2, you will have something like this. That is your answer. So answer is this is going to be sp, right? So answer will be sp, sp2, sp3 is the correct answer, right? So that is the correct answer. Okay. Next question is, which of the following pairs of d orbital will have electron density along the axis? Along the axis, along the loops, you have, along the axis, you have this d z square and d x square minus y square. Right? So answer is B is the correct answer here. Next question is, a given nitrogen. So now this is a very, very interesting question of organic chemistry. Let us see that. Right? Let us see that, students, everyone. Question is, a given nitrogen containing aromatic compound A reacts with SnHCl. Okay, it reacts with SnHCl. That means the reduction is happening in this case. So when it is getting reduced, nitrogen containing compound you have followed by H2. That means definitely this is going to be, see, if you have done enough reactions, you will get to know that this will be aniline only. Why? After the reaction of tin and HCl, if you have aniline, because after, if they give you HNO2, that means they are talking about diazotization. That means they are talking about this will undergo diazotization to give an unstable compound B. This unstable compound B, because why I have written benzene here? Because we know that in the options there is benzene. That means there will be benzene in the A also. B on treatment with phenol produces beautiful color, that is azo dye. So with phenol, it is going to give you this kind of dye, N2. Can you tell me the color of that dye? Anybody, color of that dye? They are asking you the compound A. Compound A is going to be NO2, because this will, after getting reacted with SnHCl, it will get reduced to NH2, right? So here answer is A is the correct answer. Now what is the color of that dye? With phenol, it forms orange dye. Okay, with phenol, they can also give you a one more hint here, but that will not be of any use to you, that what is the color of that dye, because that will not help you to answer. They can give you that reacted with some compound to give orange dye. Then you can say that definitely that compound to which this reacted is phenol, right? Okay. Next is the correct order of strengths of the carboxylic acid. So order of strengths, if you see here, right, we know that we need to see the stability of carbon ion here. If you see this oxygen, this is electron negative. It will try to take out, because we know that there is BARHI. Back bonding is not there in any of the cases. Aromaticity is not there. Resonance is not there. Hyperconjugation is not there in the negative, like carbon ion, there is no hyperconjugation. Rest, one effect that we are left with is inductive effect, right? So now if you see in the inductive effect, what is going to happen, students? Here you have oxygen is very far away. Here oxygen is very near. Oxygen is good for carbon ion or bad? If you have something negative, if it takes out electron, if you're under stress, someone takes out stress from you, you will feel happy, you will feel stable, right? So it will feel stable here, right? So that is why we need to have something which is taking out the stress. Here oxygen is very near and it is taking out the negative electrons from this side, right? So that is, it is going to make it very stable. So B is going to be maximum acidic, right? That means 2 is going to be maximum acidic. Here it is far away, but still there is some effect. So after this, you will have third. This is having no effect, so it will be minimum. There is least acidic order, it will be least acidic strength, it will be. So answer is A is the correct answer here. Next question is the molar conductivity of 0.5 solution of AgNO3 with electrolytic conductivity of this much at this. Okay. So molar conductivity is equal to, if this is given in centimeters, this will be kappa into 1000 divided by molarity. Kappa, this is the electrolytic conductivity, that is given to be 5.76 into 10 raised to power minus 3. This is 1000 divided by molar conductivity of this. Wait a minute. Molar conductivity of this much solution. Okay, they have given you mole per liter. This is concentration. They have given you molarity, right? So this is 0.5. So with this, you can easily calculate the value of molar conductivity, right? Okay, using this formula, you can easily get the answer for molar conductivity here, right? Next question is the relative stability of +1 oxidation state of group 13 element follows the order. +1 oxidation state, we know that is the lower. What is lower oxidation state? Let's say you have Ns and Np. Let's say you have X electron, here you have Y electrons. Lower oxidation state means Y electrons. Higher oxidation state means X plus Y electrons. Okay. Now, let us see this here. The relative stability of +1 oxidation state of group number 30 is going to be what? So, down the group, the lower oxidation state will increase. So, it is BALGAINTL, ALGAINTL, ALGAINTL. That means D is the correct answer. Always, this is always like this. Lower oxidation state, because if you talk about NS2, NP1, lower oxidation state is number of p electrons, that is 1. So that is why it will increase down the group, right? Okay. Next question is the order of reactivity of the following alkyl halides towards SN2. Now SN2 means it will be a one-step reaction. It will have transition state. It will have transition state. Is it? Now, the order of reactivity of following alkyl halides for SN2 reaction, they are asking you. See, simple thing is, as you go down the group, the length of C and I, C and if you do the, go down the group, this is CF, this is CCl, chlorine, chlorine, bromine, and this is iodine. Down the group, this length is increasing, right? Down the group, length is increasing. It is easy to break the bond. And when you talk about, let's say here you have Rx. So whenever you have a transition state, here you will have X is going to leave and nucleophile is getting attached here. That means more the strength of, better is the leaving group, more will be the rate of reaction. So this should be a good leaving group. This should be a good leaving group. For this to be a good leaving group, if you say you and your friend who are like holding hands like this, it will be very difficult to break your bond. If you are holding hands like this with this finger, only with this like this, you are holding your hand. It's very easy to break the bond. Similarly, Ri, it is very easy to break the bond. It is a good leaving group. So that is why strength will be D is the correct answer. Okay. Understood? Next is benzaldehyde on reaction with acetophenone in presence of sodium hydroxide solution gives what? Anybody? This is aldol condensation. Let me tell you why. This is benzaldehyde, that is benzene. This is C double bond O, this is H. And here you have acetophenone. This is benzene and this is C double bond O and this is CH. Now here what I'll do, I will have, there is only one alpha carbon here, that is over here. So what I'll do, this is C, this is double bond O, so I'll write it as CO. This is CH2 and I'll write hydrogen here. Because I write alpha hydrogen as different part. And here I'll write just simple CH2. Now I will connect these two things together. That is this carbon, carbonyl carbon with the alpha carbon. So this is benzene. This is the carbonyl carbon. I can also draw it like this, okay? let's say this is the, I can also write CHO like this, okay? It doesn't matter. It doesn't matter. Just for my convenience, I'll write CHO here, right? Now I will say I am connecting these two carbons. Ma'am, why? How can you draw CHO here? Array, symmetrical it is, doesn't matter. So this is C, this is CH2, and this is CO, and this is the benzene, right? I know that if I write CHO here, can I also write it like C double bond O, this is H. It is connected to this one. I know that the carbonyl carbon is getting converted, it is getting OH here, right? Rest of the things are same, right? This hydrogen is shifted here. Now what I'll do, there is dehydration from, this is alpha and this is beta position, so there is dehydration. That is why you have the formation of this double bond. And hence you have C6H5C double bond, right? Here you had only one hydrogen also you had here, no? So C6H5C double bond C, CO C6H5. C6H5CH double bond CH CO C6H5, that is A is the correct answer here, right? Those who are still confused on the concept, I'll really advise you, don't see the mock test completely blank, okay? Our team has done 2 days revision session where we have completed everything. You can go and check that out. That happened this Tuesday and this Thursday, right? So you can check that out, okay? On the live section, you will see class 11, full class 11th and full class 12th also, right? Next we have is on the basis of following thermochemical equation, this is the equation that is given to you. They are asking you the value of X for this final reaction will be. This is based on Hess's law. Hess's law. If you see this law here, you have to add all of them, right? If you add this H2O, this and this will be cancelled, right? CO and CO will be cancelled. Then carbon here, carbon will stay as it is. Hydrogen will be cancelled, right? On the opposite side they are, right? This O2 will get added. O2 will get added as O2 and here you will be left with CO2. That means you have to add this first equation, second equation and third equation and you will get your answer of delta H is equals to plus of 131 minus 282 because here it is in minus, here it is in positive. Here it is in minus, so it will be minus 424. You will get your answer in kilojoules here. Okay? Understood, students? Next is for a reaction is given to you 2A to B plus 3C, right? At a given temperature, let's say Kc 16 is given to you, what must be the total volume of the flask if the mixture of 2 volume each, okay. They are saying this is this reaction, right? B plus 3C, right? Now, I have even told, in that session, I have covered even difficult questions in this, right? So that session is very important. At equilibrium, this is 2, this is also 2, this is also 2. Concentration, they are saying the volume, let's assume the volume as capital V. This is V, this is V. We know that the value of Kc is going to be C concentration cube divided by, multiplied by 2 square, is it? No, 2 square, it will be simple 2. Okay, divided by A square. So C is what? 2 by V cube. This is 2 by V and this is 2 by V square. So this is 4 times. So this will cancel this. It will be 2 by V whole square. So answer will be 4 by V square. Right? For a reaction at a given temperature, Kc is given to you. They are asking the liters. So we can say Kc is equals to 4 by V square. So V square is equals to 4 by Kc. Kc is given to be 16 here. So it will be 1 by 4. So V is going to be 1 by 2. Okay. That is 1 by 2 liters. So answer is going to be B is the correct answer here. Okay. Understood? B is going to be the correct answer here. Right? Next is the rate law for the reaction. This is the reaction that is given to you. Rate law is given to you. If the concentration of A is half, R is equals to Ka square B square. Okay. So here what they are saying, that here you have Ka square. A means what? The rate law for the reaction, Ka square B square. The concentration of A is half. A we will half it. B is doubled. Okay. So what will happen here? This will be K. This will be 4. This is A square. This will be 4. This will be B square. 4 will cancel. That means it will remain the same only after the reaction as well. So that is, it is going to remain the same only. Right? Okay. Understood? Number of stereo centers present in linear and cyclic structure of glucose are respectively. Number of stereo centers, beta, they are asking you. So number of stereo centers in linear and cyclic structure of glucose. Linear and cyclic structure. So first you have to know the linear and cyclic structure. If you see the linear structure, there is chiral center is what? Stereo center means either it should have double bond or it should have chiral carbon. Double bond is not present, which shows geometrical isomerism. So we look for chiral carbon. So this is 1, 2, 3, 4. 4 chiral carbon means which are sp3 hybridized and which have 4 different group attached to it. So this has 4. And in the cyclic structure, it has 1, 2, 3, 4, 5. So answer will be 4 and 5 is the correct answer. Right? Okay. I think this is the last question of today, is it? This is the last question. Which of the following molecule is least resonance stabilized? Least resonance stabilized, students. Least resonance stabilized. If you see this is definitely benzene is resonant, double bond, single bond, double bond, single bond, double bond, right? Double bond, single bond, double bond, single bond. This lone pair is not involved in the resonance, right? So it also has the same resonance. This lone pair is involved, this lone pair is not involved. Double bond, lone pair, double bond, single bond, double bond, lone pair. It is also resonance stabilized. If you see this, double bond, single bond, double bond, single bond, single bond. So this part does not have resonance. So that is why this is least resonance stabilized. It is only resonance, double bond, single bond, double bond, single bond does not. It has overall resonance. It has overall. So answer will be A is the least resonance stabilized. Okay. That's it for today. Questions of your chemistry part of your NEET mock test. And students, there is some very important news for all of you that we are giving you totally free of cost. We are giving you test series, mock test, notes. We are giving you important short notes also. We are giving you marathon here for your revision, right? That is Revise-a-thon marathon that is going to happen here. That is happening from 16th to 29th of April. This is totally free of cost, 14-day free revision sprint for NEET 2025. You can enroll yourself down in the description. You will see, click on that and enroll yourself for that. This is totally free of cost. Okay. Next is Vedantu's free NEET 2025 champion test series. That is also totally free of cost. Click in the link in the description and join your series and start giving the test right now. Then you have NEET 2025 mock test. Okay. Test series is you can give it anytime. Mock test you will have to give on that particular time only, right? Okay, and you will get your all India rank as well in the mock test. Full analysis will be there, and this is only you have availability from 16th to 23rd of April, right? Okay. Next is your free NEET 2025 study buddy. Ma'am, how can we get that? Click on this QR code and you will get free NEET buddy, which will give you 24/7 smart study assistant, instant access to daily study material will be there. You just need to scan this particular code here that is available to you. Okay, that's it, students. Now I'll take your leave and I'll ask the teachers to join in. Till then, bye-bye, take care. This is Diksha Kaushal signing off. Just give me 1 second here. Okay, chalo, now I'll take your leave. Till then, bye-bye, take care. And don't forget to hit the like button, students, and do comment down here, right? What I want you to comment down over here. Hashtag NEET 2025. Okay, that is your target. Hashtag I can. I want you guys to comment it down. I can in the comment section. Okay. So this is Diksha Kaushal taking your leave. Bye bye. Taking all the best for your upcoming mock test and your exam. And do click on the description. Okay. In the description, you have all these things. So I really want you guys to enroll for the mock test and tell me the results, because that is just an exam for you, right? You will make your own strategies, right? Okay. And I will also upload some really important videos regarding your NEET 2025. You can see that on the video section that will be live today only, and tomorrow also there will be really amazing videos regarding your mock test, right? And regarding your full revisions for your next 10 days. Till then, bye bye, take care. This is Diksha Kaushal signing off. Bye bye.